Welcome to the next episode in our series of workshops on integrative organismal modeling of movement. These workshops have been sponsored by the National Science Foundation Division of Integrative Organismal Systems to help generate discussion and collaboration between experimental and theoretical scientists, and to help the community make effective use of analytical tools for understanding organismal biology. These workshops are recorded and shared as an online resource hosted through the UCI Center for Integrative Movement Sciences, so you can go ahead and browse that website for previous episodes and keep an eye out for future workshops. Today's workshop is on modeling animal locomotion in natural environments. And with that, I'll pass things over to today's presenters, and our first speaker is Glenna Clifton.
Welcome, everyone. Thank you so much for joining us for today's workshop on modeling animal locomotion in natural environments. Again, my name is Glenna Clifton. I'm a professor at the University of Portland, and today's workshop is going to be separated into kind of 3 different modules. And I'm going to handle kind of the first little less than an hour of, for the first module that I'll be presenting. Which will all be about kind of why we should film animals in natural environments, some of the considerations, technical considerations to do, as well as figuring out how to calibrate kind of a stereo camera system. And so this will include both some discussion just with a presentation, but also some hands-on walking through some of the coding involved. And so at that point, I will kind of walk you through how to get those materials in order to really be able to walk through the interactive workshop with us. But first, let's get started. So, why observe animals in natural environments? Well, there's a lot of reasons to observe animals where they actually live. And one of the main ones is that a lot of animals just can't survive in lab conditions. And so either they're just too big, they're too mean, it's too dangerous for us. They're too endangered, they're too afraid of humans. There's some animals that just actually die when they're in captivity. So to the very extreme, there are many animals that just don't survive in lab conditions. And so, for example, I studied a loon for loons for some of my past research, and they're so afraid of humans, it stresses them out so much that they die in captivity within 3 days. So there can be a huge extreme of animals not surviving in the lab, which can really limit the species that we're looking at. And so I'm sure you know many of the model systems that we tend to gravitate towards, whether it's rats and dogs or guinea fowl turkeys, bluegills, lamprey, knifefish, kind of these standard model systems, fruit flies, cockroaches, kind of the standards are partially standards because they survive so well in the lab. But just because an animal can live in the lab doesn't mean that it's behaving naturally in the lab. And so there's a lot of factors that can contribute to why an animal might not actually be behaving naturally in the lab, even if it technically can survive there. So I've kind of separated things out into a couple of different categories. The first is abiotic factors that influence how an animal may not really be behaving naturally. These are things like light levels, temperature, noise from either stuff in your lab space or kind of like environmental noise, pollution, things that may influence their habitat, like the salinity, the pH of the water if they're an aquatic species. All of these factors can really influence how an animal behaves, even though it may not be the focus of what you're studying. And there's also biotic factors. So for instance, if there's the presence of other conspecifics, other animals of the same species present, that can really change how an animal behaves and with interactions. The presence of the experimenters can have a big influence on how an animal behaves. And all of this can kind of go into the motivational state. I like to say, I know I walk differently when I'm very tired versus when I'm awake and well rested. And it's the same for animals. Maybe we can have discussions about how consciously that is true, but there's definitely variation based off of motivational state. And we know that animals have very strong motivational states. Body condition, so how well fed they are, how much fat they have on them from living maybe even an entire life in the lab. And things like having a natural versus a lab-based diet. How many animals actually get unlimited food all the time for themselves? And what does that do not only to their body, but also to their motivational state and their behavior? The last kind of category I wanted to mention was temporal considerations. A lot of times we study animals and we say, this is how they move or this is how they behave, but we're only looking at them really in a single snapshot in time. But animal behavior really varies temporally, so that varies throughout the day, and it also varies seasonally. It varies throughout their life, and it also varies kind of in a short temporal amount with fatigue, though I think we do tend to consider that a little bit more frequently in our studies. And I have two kind of anecdotes to share about this. So I was studying for diurnal behavioral variations. I was studying cockroaches, and then COVID hit. And so I ended up bringing the cockroaches back home and doing the experiments like from my living room, from my bedroom, was this whole thing. And what I noticed when I brought the cockroaches home is that they suddenly started getting a lot more active at about 8 or 9 p.m., which is never when I performed experiments when they were at work. And so our workday is not necessarily the same as the animal's workday, and that can have a big influence on the animal behavior. Seasonality is also big. And my little anecdote about this is I was studying some ants, bringing wild colonies of ants into the lab and looking at how fast they're walking. And I did an experiment and I realized actually I wanted to change some variables of it. So I completely redid the experiment about 3 months later. And what was interesting is their behavior was completely different. And so even their walking speeds, the average walking speed was about 2/3 the level of what it had been just 3 months earlier, because there's a lot of seasonal impact of the colony dynamics within those ants. So these are really important factors, and factors that we don't often report or consider. And so it's something that really helps drive us to kind of leave the lab and put the animals in their natural environment to study how they move there. Okay, so these are some of the reasons why we like to study animals in natural environments. However, it does pose a lot of challenges. And so what I'll be talking about mostly today is recording the kinematics of animals as they're moving in their natural environments. So kinematics, it's a fundamental measurement for quantifying animal behavior. Really, it can be used as the basis for kind of extrapolating to lots of other different types of analysis, from looking at the gaits and the phasing of the limbs, if it's a limbed organism, looking at the motor power and the motor coordination, the sensor motor control, and also looking at kind of like ethograms, these behavioral diagrams, kind of quantitatively looking at how these behaviors emerge. There's lots of different techniques used to record kinematics, from radio tagging, which can be quite spotty depending on how large of an antenna you have, GPS, which also can have different levels of resolution to it, inertial measurement units, also known as IMUs. These often these are great and they're being utilized a lot more. One of the challenges of these, though, is it's really restricted to larger animals at this point, because they can be so weighty. They can have so much mass for a smaller animal. There's also video recordings looking at 2-dimensional kinematics and video recordings looking at 3-dimensional kinematics. These are kind of the main techniques that we kind of measure the kinematics. And really what we'll be focusing on today, though, is the 3D videography. So for the rest of today's workshop, we're really going to be focusing on using 3D videography in order to quantify the kinematics, which help us get to all these different types of analysis, from looking at behaviors to looking at gaits. Okay, so some of the challenges, though, when you're working in natural environments is finding where the animals even are. It sounds silly, but it is a huge challenge for anyone who has really worked in the field. And so here are some suggestions. This is going to be kind of like a tips and tricks section for y'all, the next couple of slides. So to actually find the animal, reaching out to the local fish and game agencies, naturalists at museums can really be a huge benefit. It's hard to know where animals are unless you are in the place itself. So especially when you're working from afar, it can be really helpful to make contact with local experts about this. And that can be through kind of organized agencies. It can also be through less organized groups like local hobbyists working with birds. This is super easy because there's always a really strong network of birders around, but there's lots of hobbyist groups for almost every animal group. So reaching out to those individuals can be extremely helpful. One of the benefits of kind of community-oriented science and data gathering with our computers that live in our pockets these days is having apps on our phone or our web-based apps that help document sightings of different animals. And so kind of these community-guided sightings databases can be really helpful. Things like iNaturalist, eBird, the Herc Mapper, and they're usually kind of specialized based off of the animal group you're looking at. And also reaching out to other researchers. And so really kind of knowing where to go is a huge part of the battle, how accessible it is, what places might be best, or, those birds used to be here, but they haven't been here in several years, or now it's more popular over this, there's higher densities over here. That kind of information is hard to find online or published. It's really helpful to kind of make those contacts. And I just want to take one quick mention to say that one of the biggest challenges of doing things in natural environments is that it may require some permitting, which can become quite challenging and restrictive in terms of how long it takes also to get those permits. So that's something to be aware of bright and early in your process here, to really make sure to know kind of what kind of permitting you would need to be filming animals in there, or even accessing the environments. Okay, one of the major challenges of filming outside is that it's outside. That brings with it some kind of massive challenges in that your cameras are then exposed to heat, cold, and also weather, precipitation, and humidity especially. So here are just some ideas of how to kind of help. Generally, the heat is going to be worse for cameras than the cold. Cold can kind of restrict cable performance, though insulation is generally fine, and cameras themselves produce quite amount of heat, so they kind of self-warm. But in hot conditions, cameras can overheat really quickly, and it's a really big challenge. So some kind of like homemade options, you can use a Peltier cooler, which is basically a thermocoupled way of cooling, sending heat also through a heat sink there, you can see. Even just a USB powered fan can be really very portable, using kind of like a phone power bank, and run for quite amount of time. Precipitation and humidity can be a challenge. Oh, the other thing I wanted to mention with heat is having little portable structures. You can see here a little, what I call a little pup tent that I set up. I think most people use it for children on beaches, but hey, I used it for high-speed cameras by a lake. And so that can really help both with preventing kind of direct sunlight, as well as helping kind of visualize things on computer screens, because glare from the sun can be a big challenge for that. For precipitation and humidity, there are some cameras that are pretty robust, especially the consumer grade cameras like GoPros that have really robust cases for them. And some of the higher end high-speed cameras also work with some kind of custom camera builder or camera case builders to help kind of have more waterproof housing. That's not always the case. Here I'm showing an example of actually a fully waterproof case that I built personally for my own camera that you can see, or the cameras I was working at, that you can see in the right of this picture. And so there wasn't a case made for these, and I wanted to submerge them underwater, which is probably more weather than most cameras will see if you're just not actually submerging them. And so I just used a Pelican case and kind of built in my own structures, including cable hose for the cabling. On the side that you can't see is a plexiglass panel that I cut and attached on with some silicone aquarium adhesive. And so you can actually make your camera systems quite weatherproof. Did a lot of testing before I actually put the cameras in these camera cases and submerged them underwater though. So here are just some suggestions or tips and tricks for kind of making your camera system a little bit more robust to the weather you might find out in natural environments. Okay, so some backwards design is always a good place to start if you're thinking about doing a recording session out in a natural environment. So some things that you may want to think about is, first of all, what recording rate do you need? How, a lot of times I go from what the frames per stride is. So if I know an animal is performing a certain behavior, or per cycle, shall I say, if it's not walking, but how many cycles is it doing per second? And then how many frames do I really need per cycle? I usually aim for around 20. At least. And then the horizontal field of view is a really important one. As animals are going to be behaving naturally, there's more variability in where they might be. And so knowing your horizontal field of view, which we'll talk about calculating in a few slides, knowing the depth of field of what you need to be in focus of where the animals may be. Lighting considerations are going to be really important in terms of like, when are you recording this? Is it going to be in bright sunlight? Are there going to be glare reflections like off of this lake that I had, or are they nocturnal animals? How much access will you have to power? Are you going to have to power everything by battery, or will you have access to some kind of power supply? And how are you going to position your cameras? What kind of restrictions are you expecting for the terrain that you're on? So I'll talk more on that later too. So these are some of the things I like to think about first, and then kind of work my way into designing my camera system and my setup. So some considerations for your cameras. Well, there's kind of 3 ways you could go with choosing cameras. You could go for the consumer kind of GoPro-esque cameras. They're usually pretty inexpensive. However, they often have a lot of wide angle, which leads to a lot of distortion. But they're very portable. They've got spare batteries. They've got a lot of accessories. They're really light and easy to mount places. They can be kind of difficult to trigger and especially to synchronize. There are some tools now that really help synchronize these cameras to each other, including using like audio to really synchronize within one frame. But it is a hacked synchronization and not a proprietary from the company synchronization. And a lot of times they tend to overheat. They don't usually include internal fans. But you can really put together a large kind of army of these GoPro-esque consumer cameras. Then you've got your specialized high-speed cameras. I've listed just a few of the companies out there that make these. They're usually much more expensive, but they come with a bunch of features, everything from really easy to trigger and synchronize with each other to automatically triggering if it notices something that's in the field of view. They tend to have kind of the most important part, faster frame rates, higher resolutions, and they tend to be more light sensitive. And you have a greater variability of lenses that you can attach to them, which make things really different. Then recently, kind of in the last maybe 5 years or so, there's been kind of a middle ground option. Two of the main companies for these are Chronos and Firefly. They are kind of this mid-range level of camera. I haven't heard a ton about these cameras yet. I definitely know that some people doing kinematics use them. What I have heard is that they don't have the best like customer support or technical help, as you might find in the specialized high-speed cameras, which have a lot more kind of technical support there for you, and that they tend to be more difficult to synchronize. But they're definitely something to keep your eye on, and I also would love to hear if you have experience using them. They are an interesting kind of route to go. Okay, so when you're choosing your camera from any of these kind of 3 options, some considerations to think about when you're choosing is how are you going to control this camera? The consumer-based cameras tend to have controls on board with a touch screen or some kind of viewfinder on the camera itself. Most of the high-speed cameras have to be plugged into a computer and visualized through a computer. This poses challenges because then you have to bring a computer with you also into the field and power that computer in the field. And it also makes it difficult to kind of see what your camera is looking at. So if you're recording here, but you have to look at your laptop over here to kind of see what the camera is seeing, that can make things kind of more difficult in terms of positioning and quickly repositioning your camera. So having a viewfinder of some sort, even if it's kind of a digital viewfinder or just a screen showing what's on the camera. Pixel size can be really important. A lot of times cameras will tell you about how many pixels they have, but not the size of the pixels themselves. And the size of the pixels is actually one of the first things I look for in a camera. Basically, larger pixels collect more light, so they're going to be more light sensitive. And so especially if you're considering recording in low light, pixel size is going to be an important measurement for you to consider. How are you going to power your cameras? So again, a lot of these cameras must be plugged into a computer, especially for the high-end, high-speed cameras, which means that you're going to have to bring a battery with you. There are some kind of portable battery stations including ones that have solar attachments to them, or you can use a marine battery, which is how, for the cameras that I showed against the lake there, that's how I powered those, is I had a marine battery that actually was on wheels, and I would wheel it out to the lake edge and plug the computer and cameras into that. There are some cameras that have onboard batteries or also external batteries, and I'll show you the cameras that I use that have those. Storage, how are you going to store that data? If you're planning on portable in the field recordings, again, you might not want to bring a laptop with a hard drive with you. And then how are you going to save those files? Are you going to have enough storage if you're just bringing a laptop to store all of your really large high-speed videos on them? Or can you buy a camera that has internal storage for it so that you only have to do a download in the evenings after you've recorded? That brings up another point of how long it takes to save. A lot of times when you're looking at natural animal behavior, it's either on or off. And when an animal is in the field of view, you want to be recording it as much as possible. And it can take a long time for some cameras for that video to actually save to the camera so that you can rearm it and re-record. And so having kind of like live recording modes where it can save and record at the same time is a really helpful feature for naturally behaving animals. Triggering and synchronizing can be quite tough in natural environments. Really long BNC cables, or figuring out how you're going to get your camera system triggered and synchronized. So thinking about how a prospective camera system does that. And a last part is, if you want to calibrate your field of view for more quantitative kinematics, you're going to need to calibrate it with the cameras held still relative to when you are recording the training data. And this can be quite challenging in natural conditions when there's wind or rain or just people moving around. And so investing in really good tripod heads, I prefer ball joint heads, as well as pretty steady legs is going to be helpful. And then also thinking about having some like ballasts for your tripod legs can be really helpful. Okay, so these are all things to consider. Let's keep going in our things to consider for your setup. Choosing the right lenses can make a huge impact on your ability to film in natural environments. One of the big things to consider is calculating what field of view you're going to need. So how much space are you hoping to record, which is especially important if your animal behavior is unpredictable. You want to have as much view, but also as much resolution to see the detail that you need. So it's this trade-off between that. When you have figured out what field of view you need, here are some calculations to kind of know for your camera what focal length lens you would need. It's really based off of the sensor size. A lot of times cameras don't actually tell you the sensor size, but they do tell you the pixel size and the resolution of the sensor, and therefore you can calculate the sensor height and width. And then also the lenses that you're buying may be listed as a certain focal length, but that might not be the effective focal length of that lens on your camera system. Most lenses are listed with a focal length for a 35 millimeter, also known as a full frame sensor. And so if most high-speed cameras have a smaller sensor than this. And so that induces something that's called a crop factor, which effectively means that for a lens, say that you had a 24 millimeter lens and you had a crop factor of 2, that lens would actually act, did I say a 24 millimeter? It would act like a 48 millimeter lens then. So it would actually double the effective focal length of that lens, which means you would see a smaller portion of the world view. It would also mean there would be less distortion. So there's kind of interesting things there. And to add to the complexity, some camera makers make lenses that are intended for smaller frame cameras. So for instance, Nikon has their DX series, Canon has the EFS and EFM series. And so these lenses are actually labeled for a smaller sensor. So it can be quite challenging to figure out what lens you actually need. But here are some equations to help you in those calculations. Okay, depth of field is another really important thing to consider, because your animals moving in natural environments are probably going to be more unpredictable. And so you want to be able to have a larger range of the view in focus. So here's how you calculate depth of field. It depends on the focal length, your aperture or your f-stop of your lens, the distance to the subject, as well as something that's called the circle of confusion. I won't go through what the circle of confusion is in too much detail, but basically it's like the acceptable amount of blur, how much can be considered kind of like in focus in 1 pixel. And generally for small sensors, C ranges from like these values 0.01 to about 0.02. And basically the main thing to think about here is you want a large depth of field. And so to do that, having a small aperture is better, which is good, but there's a trade-off, but then it lets less light into your camera, which if you have large pixel sizes is maybe less important. If you have really good light sources is less important, but it is that trade-off between those. You can have a short focal length, which could increases your depth of field, but then there's more distortion. So again, there's a trade-off there. And you can also have a longer distance to your object. But again, remember that you're also trying to figure out what size you want that animal to be in your field of view to get the resolution that you need. So all of these features have trade-offs, and it's quite actually, I think it's actually quite a complex process to kind of figure out the trade-off between these different factors to figure out what your system should look like. Some other things to consider when you're purchasing lenses. Generally, you always want to find a lens that has a manual aperture control. So again, this is mostly for those higher end cameras, the GoPro consumer level cameras. You don't have to think about this. Their lens is already built in. But if you are choosing an external lens, most high-speed cameras do not allow you to do the automatic aperture control that DSLR cameras, the kind of what they were made for, enable. So you're looking usually at older lenses that have the manual aperture control, which can be beneficial. They're usually cheaper. And you can usually find used versions of them that are quite lovely. You're also probably going to want a manual focus. There are some high-speed cameras that do some automatic focusing, but generally having the ability to manually focus is a good way to go. And you're going to have to decide whether you want a prime lens or a varifocal, also kind of colloquially known as a zoom lens. Prime lenses, if you can do it, they come with a whole lot of benefits. They're optically better. There's less distortion, both from chromatic aberrations as well as kind of barrel and wide angle distortion. They tend to be much cheaper, lighter. However, they're just less versatile. So I think a lot of individuals who are doing research in natural environments tend to go for a varifocal or a zoom lens. But if you have a pretty well-defined natural environment setup, if you're able to go for a prime lens or even a few prime lenses can be really helpful. It just doesn't enable you kind of the really quick response time and the flexibility in changing those lens parameters on the fly. Okay, few more tips and tricks before we take a break and then go on to some interactive workshop components. Camera placement is really important for your ability to reconstruct the three-dimensional kinematics. There's a whole paper that goes into a lot of great detail on this listed here. But generally, you want to place cameras farther apart to increase the reconstruction, like the calibration efficiency. But if you're too far, then you don't have as much field of view overlap. So again, it's a trade-off there. Adding extra cameras doesn't actually improve your calibration efficiency. However, it can help if there are occlusions or multiple animal tracking where there's overlap. It can kind of help resolve some of those confusions. Generally, you want to aim for around 30 to 60 degrees of angle between cameras, but it really depends on the field of view also of the camera, so kind of like what lens you're using, and you want to avoid kind of directly parallel cameras because there's not a lot of overlap, and also perpendicular cameras because it's harder to see the same body points in those. So slightly less than that would be, or more than that would be ideal. Lighting, I always think, is actually probably the hardest part of your setup, especially if you are planning on doing automatic tracking. consistency is going to be key. The more consistent of an image that you can send into an automatic tracker, the easier job you are giving it and the better and more reliable your results will be. High contrast as much as you can between the subject and the background. That may be informing where you place your cameras. Maybe it's filming birds against the sky instead of against a rocky backdrop. Auto exposure or auto lighting features are generally included in your cameras, and you would like to avoid them, mostly because either they may change settings so that your subject is actually blurry or induce more pixel noise, or actually to some extent sometimes even change the internal parameters of the camera so much so that your calibration isn't relevant to your recordings. Low light situations are especially challenging, and that's because it requires a larger aperture, which is listed as a smaller f-stop or an f number, and that really can reduce the depth of field. So if you need really an open aperture, then you have a very narrow depth of field, which is hard for naturally behaving animals. So I just wanted to show one of the recent solutions I have. While this setup is currently in my lab, it's certainly field portable. You can see here kind of my 3 camera system. These are Fastech TS5 cameras. They have touch screens on them, as well as the internal batteries and external batteries, and as well as internal storage. And these strobe lights here are actually infrared strobe lights that attach to the cameras themselves and synchronize with the sync out signal from the cameras, so that there's consistent infrared lighting. And then the cameras themselves, I can put kind of an infrared pass filter so that only the infrared light is getting into the cameras to make the lighting really consistent. So that's one way that I've tried to solve this. Other camera systems have lights that LED arrays that they work with. So it's something to consider when you're thinking about your setup. Okay, so this concludes kind of the tips and tricks of things to think about when you are setting up your camera configuration for filming in natural settings. I figured I'd just take a minute or two or answer some questions if you have any, or comments if you have any suggestions. I think we have a wonderful level of expertise in the attendees, so feel free to share them. We'll just take a minute before we move on to the next section. Okay, great. So there's a really good question about how do you test the quality of the synchronization? And it's a really good question. Thank you for asking it. Generally, what I do is I put a blinking LED into the field of view. And so you can either do that, for instance, like an Arduino has a light, an LED on it, and you can have it blink at certain frequencies that you can set it. Or even you can just turn on an LED sometime in your recording. So that's kind of the easy way, or even like a laser pointer or something. But if you have some kind of light source and you turn it on at some point in the recordings, you can then scan throughout the recording and see when the light turns on for the first time. And so usually I will test my synchronization that way by having some kind of light turn on and off. There are other signals that you can use, audio signals, even just like dropping something or seeing when 2 things collide. I think that the light is the easiest one to see personally. Yes, and you can, it depends whether you can. So there was a follow-up question, is if you're able to detect, to automatically detect this event. Usually not. I usually do this kind of when I'm setting up the camera system and then assume that it's fine throughout the rest of the setup. I do this like before we're out in the field, and then I trust the system after that. I do it enough times that I gain my trust. You could do something that you can write a script to automatically test when a light appears. Usually what I'll do is I'll kind of close the aperture really low to make it a very dark scene, and then turn on the LED so that there is a very easily detectable moment. And you can absolutely make some like scripts to automatically detect when that light appears. But as far as I don't automatically detect it, I usually tend to do this before I'm out in the field, and then I just trust that we have figured that out for them. Yeah, and there was a great, I don't know, can people see the comment from YT? If not, I will read it out. That pretty much anything under 1,000 frames per second, you can achieve similar data collection from all 3 levels of camera, but the less money, the more technical know-how you need to have, and the more confidence you need to have about the instrumentational skill of the user. So he's used all 3 levels of cameras in his lab and in the field. Great. Okay. So the next stage of my presentation today is going to be talking about calibrating your camera system. And so for this, it's going to be a bit hands-on. We're going to be using some Python to calibrate our cameras, both their intrinsic and extrinsic variables of them. And so there will be a time where I ask you to download some materials if you would like to walk through it with me. So I will send that link to you when we get to that point. But first, some background information, just making sure I have the link for when we need it. Great. Camera calibration. So the workflow I'm going to go through here is generally the workflow that I use, where you record a calibration object. You find the intrinsic variables that quantify basically the properties of your camera system, the camera plus its lens. That quantifies both the distortion as well as kind of how the light moves while it's in the camera. Then I look for the extrinsic variables. So this is basically how your cameras are oriented relative to each other. And then lastly is, I don't really necessarily want to know how the animal is moving relative to this one camera. I want to know how it's moving to the world. And so it's then kind of rotating all of that relative to a world coordinate system that I have filmed. Okay, so first let's talk about recording a calibration object. There's lots of different calibration objects that you can use. Kind of the 2 main ones are looking at a wand, some object of known size, where there's differentiable parts of that wand that you film. You can do and analyze your wand calibration quite easily in things like MATLAB, using EasyWand or Argus, or in EasyWand using MATLAB, sorry, or Argus for kind of more consumer grade cameras. Generally, you're going to need hundreds of frames. But one of the good things is that once you then track the animal subject, you can also use those frames for your calibration. So it is a little bit less daunting than it originally seems. It must be rigid and of known dimensions, and you have to figure out how to get it into your field of view. So here you can see an example from this paper where they used a rod, part of the rod of a fishing pole, and they threw it into the field of view, which is great. I've also used undergraduates that used an inflatable rowboat to wave a wand in the field of view. So you gotta have to figure out a way to get the wand in the field of view. That's one of the big challenges. And then kind of track the 2 sides of it. Another way, and it's kind of, I think, an easier way, if you can do it, is using a checkerboard. Or there's actually a bunch of different patterns. Checkerboard is the most commonly used, but there's circle arrays. There's also arrays of dots. There's arrays of QR codes. There's a lot of different ways actually of making a calibration kind of panel flat object. But the checkerboard is definitely the most commonly used one. So I'm going to be talking about checkerboards here. This can be done in lots of different ways through lots of different programs. There's a toolbox in MATLAB that is very, very easy to use if you have access to MATLAB. I use Python using a package of functions called OpenCV, which are computer vision packages, and that's what we'll be using today. And the reason why I like that is because it's free and open for everyone. And also I just prefer Python to MATLAB coding these days. You need at least 10 frames. Generally, you want to aim for your checkerboard to be around 1/4 to around 1/2 of the size of your field of view at the range of interest. It does expect to see a white border around that checkerboard, so can't crop it in just right to where the checkerboard is. And you need to be careful about what dimensions you have. So for example, the one shown in this image is a 9x6. Generally, it's better to have an odd by odd because then you have distinguishable corners. And there are kind of available pre-designs of these online that you can find. There's a link in the code that we'll look at later as well. Okay, so when you're recording the calibration object, first of all, you want to keep all your camera settings basically the same. Remember, this is specific to how light is entering your camera. So if you're going to change your focal length of your lens between your calibration and your recording, that's going to need a different calibration. So even things like changing the aperture, you really want to reduce changes as much as possible. So keeping everything as consistent as possible with your camera, you record a bunch of frames of this object moving throughout your field of view. You want to try and cover the whole field of view of each camera, even if it's not in the shared field of view. And that's because we're trying to quantify both the intrinsic for each camera parameters, as well as the shared kind of configuration of the cameras. You want to make sure that you're varying the pitch of the checkerboard, not just where it is like this, but you want to put it at weird angles, as much so that your cameras can still visualize it. And you're going to need even more if you're using a wide angle lens, which tend to have lots of radial distortion on them. So any lens that's less than 35 millimeters, make sure that you have a lot of calibration frames. Okay, so say you've recorded your calibration object. What are the steps? First, looking at the intrinsic variables. Again, that's how to quantify your system, your camera and your lens. The zoom, the focus, the lighting wavelength all matter. So if you're doing this suddenly in visible spectrum light, but you're recording in infrared, that can actually change your calibration. So camera positioning, though, should not matter. So even if you forgot to do this or something happened in the field, you could come back and quantify the intrinsic variables as long as you're recreating the exact zoom, focus, and kind of lighting wavelength that you were using before. The next part is looking at the extrinsic variables to orient them next to each other. Camera positioning matters for this one. And if you change where the cameras are positioned, you have to change your extrinsic variable calibration. And then also relating them to your 3D coordinate system in the world, also camera positioning matters. All three of these that we're going to use today require OpenCV. Downloading OpenCV can be a challenge. If you are planning on doing this, here's some tips for you. However, you can also use Google Colab, which is what we'll be doing today, if all of this seems too much for you. Okay, so let's start with the intrinsic variables. So, and also if you want more technical information on all of this, this is kind of the main go-to resource for it. So feel free to check out this kind of textbook. So intrinsic variables quantify the camera properties. I'm not going to go over all of this, but you can, it's here for you to read and reference back to. There's a lot of math, basically. But what we're doing is figuring out how light gets from the world to the camera, how it gets to the 2D sensor of the camera. And so we'll be looking to calculate this matrix K kappa here, which are the intrinsics of the camera. And then the second part, we'll look at these, both the rotation and the translational variables, so the rotational matrix and the translational vector that define the extrinsic variables of the camera. Okay, so again, I'm not going to go through the whole derivation of this, but this intrinsic variable does measure on scaling factors. So basically knowing the focal length versus the pixel size, the offset of where the light directly comes in the camera relative to the sensor, called the principal point, and also the skew of the sensor. So it may actually not be a rectangle, it could be a parallelogram. So all of those go into generating this matrix. I will leave it here for your reference in the future, but I will not go into any more detail now. And I will also say that I have never taken a computer vision or engineering course in general, and have taken a single computer science course in my life, and I've still, been able to figure this out. And so if you're feeling overwhelmed by this, don't worry. I think there's so much online, and especially that book, that can help you kind of piece this together, even if you haven't had any kind of formalized training in it. Okay, so now that assumes a pinhole camera. There's a lot of distortion also. When you don't have a pinhole camera, you can see what the internals of this lens here, this 28 millimeter lens looks like. And so the other thing that we'll be calculating are distortion coefficients, which effectively tell us how much radial and tangential distortion there is. There's usually not very much tangential distortion in cameras today, but there can be quite a bit of radial distortion. Okay, so let's actually work through the example. So if you go to the Google Drive link, you can download three things. There's two Python notebooks, so the camera intrinsics and camera extrinsics, and then there's a folder of checkerboard photos that I've taken with my camera system, and you should download all 3 of them, and then go to a browser and open colab.research.google.com. You will need to sign in with your Google account and then upload the first camera intrinsics Python notebook. So here we are. If we go here, okay, so here we can download all 3 of these. I'm just doing it with everyone to make sure I don't speed ahead. Okay, so once you get to Google Colab, you can upload a file by clicking here and dragging your intrinsic file here. And then when you do, so I already have this open here. There we go.
Should we see your Colab window right now? Because I don't.
We can switch over to that. Yes. Thank you. There we go. Okay, great. Now from here, we can go to locate in drive, which will open up where this is in our drive. Now I've actually already made a folder with these in it called IOMM workshop, but you could make one for yourself. So I would suggest making a new folder called IOMM workshop and then dragging your recently uploaded into that, and then also upload the calibration images into that folder. So what you should end up with is some folder that has all 3 of these files in them, and then you should have opened up your camera intrinsics file. So now I'm going to walk through this quite rapidly, but it is pretty well commented, so it should be able for you to walk through it on your own. This is a Python notebook that has cells of code that you run one by one, in case you don't have any Python experience. And the way to run a camera code cell is by hitting Shift and Enter on your keyboard. So you can see I can hit Shift and Enter, and it will, in this case, import lots of things that I need in order to run this. And a checkbox over here says that it successfully ran. So after importing some of the stuff that we need, we're going to load this Google Drive to our document. It's going to ask to make sure that we want to do this. So you're going to have to give it access. And then it will actually mount your Google Drive so that you can upload those calibration images. And it's going to check that it worked by showing everything that's in your drive. You can see here that there is that folder that we made before, and we can then use that to get all of the calibration images from it. So we're going to tell first the notebook where that folder is, as well as some information about the cameras, the pattern size of the checkerboard, as well as the actual size of the physical checkerboard, what the size of the boxes were. And we're going to look for all of the calibration folders within the calibration files there that have all the pictures that were taken. Then we're going to find the checkerboards. So this code here is going to use OpenCV to read in each image. And then it's going to look for the checkerboard edges after we convert it to grayscale. So for each image, we're converting it to grayscale. And then we're going to do a little bit of noise reduction here. And we're going to find the corners using this find chessboard corners function from OpenCV. If they're going to find them, we are outputting them to certain variables that I have kind of pre-allocated before. And then it's going to save them to a folder so that you have kind of all of these important variables. So remember, we talked about the camera matrix being kind of the intrinsics here, that K or kappa. Then also the distortion coefficients are the main ones that we're really looking for here. And so you can see here, our code is working through. So for our camera 164, it found 25 images. It found the checkerboard corners in these images, it calculated the camera matrix, and then it found the distortion coefficients, and then it saved all the camera parameters, and it did the same thing for the other camera. Then this is just some code I wrote to visualize how good how well this worked. You can see these are just kind of two random frames where we have plotted those checkerboard corners in them. And then this is for all of the frames that were shared or for one camera where the point was that was found, and then also the reprojection of it given the camera intrinsics, where would we expect that to be as a measurement of how well our camera calibration was. So the offset is around 0.15 pixels or millimeters, sorry, for each of those because we calibrated it with the checkerboard size. So that is now how we have found the intrinsics of our camera. And I'm going to keep chugging on along. Again, this should be pretty well commented for you to look at later and walk through if you want to. I'm going to go back to the presentation very quickly. So we found the intrinsics. Next part is finding the extrinsics, which is basically how the cameras are related to each other and then relating them to the world coordinates. Again, this information is basically here for your reference. I'm not going to go through it too much, but the main things that we need to know is how the cameras are rotated and translated with respect to each other. So generally we think of this as a rotation matrix that is 3 by 3, but expanded to 4 by 4, and then also a translation column vector X, Y, and Z, but expanded to a square matrix so that you can take the cross product of the two matrices. Some other things that you're going to look for are finding the essential matrix and the fundamental matrix. This basically helps relate a point found in one camera to where it should be in the other camera. And so you can see here, if there's a point seen by this camera over here on the left, it could actually be one of several points seen by the right camera, camera 2 over here. And all of those points lie along a line called the epipolar line. And so this essential matrix basically says for a given point relative to one camera, what line must it be viewed on in the second camera? And then the fundamental matrix actually takes this one further in that it the essential matrix assumes that you've already kind of know the intrinsics of the camera, whereas the fundamental matrix incorporates those intrinsics into the matrix itself so that you are working on like from where it's seen on the sensor, where should it be in the other camera sensor instead of thinking about where it is relative to the first camera. Okay, so again, let's go back to our coding now really quickly to walk through this. Okay, so now we can go and open up our camera extrinsics. And again, I will walk through this really quickly, but again, it's pretty well commented, so you can work through it on your own, and also feel free to email me if you have any questions. So again, here, we're going to import kind of the things that we need to work on. It may ask you to confirm that you can mount the drive. I've also listed a bunch of links here. There's so much information about camera stereo calibration on the web. So here are some of the links that I found most helpful for you for future reference. Okay, again, we're going to tell it where all of the information is, those calibration images are. We're going to find where all of the folders are, and we're going to find where the information about all of the images that we have. So first is we're going to get this kind of stereo calibration, the extrinsic values. So we're going to load in all the information we have about the cameras, including the intrinsic parameters that we saved beforehand. We're going to tell which camera pair, if you have more than 2 cameras, this is important, that you're calibrating relative to each other. You're going to find the frames when the checkerboards are in both views, and you can actually downsample this. A lot of times I record hundreds of calibration frames, and I don't want to do this calculation on hundreds of frames because it will take a really long time. And so you can downsample it here. And then it's going to find, using a function called stereo calibrate, how the cameras are oriented to each other. And again, the 4 main outputs here are the rotation matrix, the translation vector, the essential matrix, and the fundamental matrix. So we can run this cell here. loading the information, it's finding how many frames have the checkerboard in both views, it's downsampling them. I decided with only 20 frames, I'm not downsampling them, and it completed the calibration of the system. We can plot out what the parameters are that it found. And then we can define something called the projection matrix for each camera. You can use something called stereo rectify, but it was giving me kind of weird results. And also I kind of prefer to do things manually. So I just did it manually, calculated down here. And so you can calculate what the projection matrix is. And then we can calculate again this reprojection error. So given we know we tracked where they are, and now given what we know about the cameras, where would that point be? And let's see what the difference is between like the actual measurement and the kind of like based off of this is where we think it should be. And that's a good measurement for kind of how good your calibration is. So this computes that difference between them and points it out for us. So the average error is quite small here. Ideally, it would be less than 1, but it depends on what units you're using. And so this is then testing out just on a single frame. And you can see that the projection error just for this frame is less than 1 millimeter, which is great. And we can plot and just see how well these are doing. Now I've used an interaction ability in Python here, which doesn't work super well with Google Colab, but would work better if you're working outside of Google Colab. And so this just takes some random frames and plots on it the points that were found from the automatic chessboard corner finding, and then replots the reprojection of where we would kind of expect them to be and sees how well they overlap. And you can vary kind of which frame you're looking at. And lastly, we can save this information. Here's just a little aside about the disparity matrix. And then we can basically orient now. So we have camera 2 is relative to camera 1, but I really want this relative to the world coordinate system. So what I do with all of my calibrations is the very first frame, I film the checkerboard where the corner that I know is the origin is at the origin. And I use that very first frame as the world coordinate system. So that's what I've done here. And this runs and it calculates things relative to that origin system. So you can see here, these are the photos I took of the origin with the checkerboard, the very first frame with the checkerboard with this white star at the origin. The red is the positive X and the green is the positive Z. And you can kind of get a sanity check here where it tells you how camera 2 or each camera's position should be relative to the checkerboard, to the origin, and then also how they should be relative to each other. And that's a really good way of just kind of making sure that your calibration is what you would expect it to be. Okay, so I've now gone over like at least half a semester of computer vision lectures in about 15 minutes. I am happy to take some questions. I do want to keep moving because I don't want to take up the time of all of the other great information. Like I said, these documents should be pretty thoroughly commented. And then also I have provided links to more thorough information about this, especially that textbook that I mentioned earlier. Feel free to reach out if you do have any comments. Maybe I have time for a question or two as we switch over to the next presenter as well. Yes, and then the last thing also I forgot, sorry, I wanted to mention, I knew I was going to forget to do this, is that once you have your camera system calibrated, you then want to track your animals, which can be hard in natural environments because you usually need really large data sets. Using deep learning trackers can help a lot here. Kind of the main one is Deep Lab Cut, but there are other ones that use deep learning, including DLT, DV8, Argus, Leap. Setting these up is can be kind of challenging, but these are very well documented. So I'm not going to go over any of the tracking here because it is so well documented in kind of like each of these cases. So, but feel free to reach out if you have any questions on getting the tracking up and working. And then just like how we reprojected things in the checkerboard example, you can do that using the variables you have. You can do that for all of your tracked data from these deep learning based trackers or manual tracking. Okay. That's what I have. Whirlwind of computer science or computer vision headed your way. So.
Thanks, Glenna. I think YT has a comment in that's only visible to the host
yes.
panelists, if you wanted to read that.
Oh, yes. Sorry, I didn't see that. Oh, right. Another great thing you can do to kind of, I call it working through your trust issues with your calibration or with your deep learning based tracker. Either way, you got to work through your trust issues. And another great way of doing that is to check by having like a 90-degree object or an object of any known dimensions through it. And so I'll often either track an LED, which is just easy to automatically track, or an object that has two LEDs on, or 2 or 3 LEDs on it, so that you can check kind of the orthogonality of those objects. But I like to do things that you can track automatically. So that's why I like to use LEDs, so that it's easier to track them without me having to do anything. So write the code. Yeah, great point, YT. Thank you. Amir, are you next?
Hi everyone, my name is Amir Patel and I'm from the University of Cape Town, all the way in South Africa. I'm really grateful for what Glenna did, and I think it sets me up quite nicely. So what I'm going to be talking about today is, once you've gone out into the field and you've collected this data, how can you actually reconstruct 3D animal motion? So my talk is about a multi-camera fusion for studying whole body locomotion in the wild. So we'll look at 3D reconstruction. Although Glenna did touch on it, I'll talk about some of the issues that can arise. I'll talk about sensor fusion, and I'm really going to try and keep the maths as light as I can. I've linked some resources at the end where you can get some really good insight into sort of more of the math stuff. And then we'll look at 3D kinematics again. Probably going to give I guess a semester's worth of dynamics, but I'll try it again to keep it quite light. And then what I think will be quite informative is I'll do an example of tracking some cheetah data, and all of the stuff is up in the folder if you guys wanna check it out. Okay, so my focus is studying maneuverability, much like Monica's, and I've chosen the cheetah as a model animal. Well, firstly because it's one of the most agile terrestrial animals, and we also have quite easy access to them here in South Africa. And the idea is that we want to study maneuverability. The stuff is straight out of Monica's one of her chapters in the book she's written about. That maneuverability is quite interesting because it has these trade-offs between energy economy, safety, time. All these things need to be traded off in order to execute maneuvers. And of course, to understand these, we need data. So that's sort of what this whole talk is how do we actually understand animals in their natural environments and collect the data to do that. So my group studies captive cheetahs mostly, and we've got a good partnership with 2 cheetah sanctuaries here in South Africa, and the ethics is pretty easy because we're not attaching anything to the animals. But yeah, as Glenna mentioned, that can be an issue. But yeah, so what they do for enrichment, just to keep the animals sort of exercising, is I think every second day they have the cheetahs chase a lure or stimulus, basically a piece of rabbit pelt on a track, and the cheetahs then gets their prey drive going, and then they chase it. So what we did was we set up 2 sets of 6 cameras out in the field. We used GoPros, Hero 7 Blacks. So those are sort of on the lower end, the consumer grade cameras Glenna mentioned. So lots of issues with synchronization and calibration, which I won't show you exactly how we solve them, but I'll give you the code and mention it, and how we can then use that to do reconstruction. So Just some videos, some examples of the stuff that we've collected. Show you over here. So all captive bred cheetahs. So they've been hand reared, these ones in particular. So they're really okay with humans and quite lazy, to be honest. Okay, so as you can see in that video, we've got lots of cool videos, but they're really challenging. So as Glenna mentioned, we had different times of day. So we tested and collected data in the summer and in the winter. So sunlight was a big issue. Different seasons. So you can see in the top left is in winter. There's lots of greenery. Whereas in summer it's quite dry. You can see over there we had different cheetahs. One in the bottom left is actually a variant of a sublineage of the cheetahs called the king cheetah. Angles, distances, occlusion. You're dealing with a wild animal, so it's really going to move wherever it wants to move. And what was particularly challenging is that most of the cheetahs didn't have any markers. So some very small subset of them, we had some stickers on there. You can use on the video, some pink stickers, but most of them were, no markers. So really tricky problem. So it's very difficult. Luckily, during my sabbatical in 2018, I met some very smart people, Alexander and Mackenzie Mathis, who are now at EPFL, and they developed a deep lab cut. And I worked with them and to extend deep lab cut to capturing 3D motion out in the wild, and that was actually on the cover of Nature Protocols. You can see a cool picture of a robot-looking cheetah. So how it works? So let's see. So you basically give it these raw videos. So we got 6 views of the cheetah, and then DLC automatically labels the points for you. You can see there. So all 6 views, and what's amazing is that it's one network that actually does all, any view, any kind of cheetah, as long as we give it the training data. But I think some of you may have seen there that unfortunately some of the marker locations, you can see some outliers there. So, sometimes every now and then it would jump off and think it was on a plant. Also, because it's being automatically labeled, it's not like, somebody actually clicking it manually. So there is a bit of noise. So you can see there, so the points move around a bit. So really tricky stuff for reconstruction. So how it works, just again, from a conceptual idea, is that each camera sees, for example, the tip of the tail over there. Then the next one sees the tip of the tail, and the third one. And then all of these can be combined with sparse bundle adjustment. So that's the usual kind of 3D reconstruction technique. I think that, things like EasyWand or DLT would have, even and OpenCV as well, as Glenna mentioned. So it takes all of, the views and the camera calibration that you've given it, and then says, if this is the pixel location in this view, in this view, in this view, this is where the most likely 3D point is. So obviously things kind of fall apart when you have noise, right? So let's say, for example, camera 2 can't actually see I don't know if can you guys see my laser pointer, but the camera 2 maybe putting it slightly off the tail. There could be outliers. So maybe camera 1 picks up a leaf on the tree and says that's the tip of the tail. And then also what's really difficult in in a more natural environment is tons of occlusion. So, you know, camera placement is important, but also, you know, the animal just may turn in a way that you can't see the limbs, or it may do a sudden maneuver or an abrupt turn and there's, you know, tons of dust or something. So really, really tricky problem. So if we just naively took, you know, a sparse bundle adjustment and using deep lab cut points, this is sort of what we'd end up with. And if you squint really hard, you can see a cheetah running in the bottom there. But it's obviously not great, right? So you see lots of noise and, you know, not quite a complete looking skeleton. So how do we get around this, right? So I'm an engineer. And one of the things that we often do when we're trying to control a robot or, you know, an aircraft or something, is we do something called sensor fusion. And another thing to think of it like is data fusion. So you have sources of data from you know, multiple sensors. How can we combine all of those in the best way possible? So why do we need sensor fusion? You know, so can't we just measure, you know, the 3D points? And of course, yes, if I could put my cheetah in a Vicon, you know, suit, or in a biomechanics lab running on a treadmill, like, you know, that would be really easy, but we can't, right? So one of the reasons we want to do sensor fusion is because of quality. So you may have a noisy sensor. So deep lab cut is awesome, you know, and I haven't actually used any of the other deep neural network kind of, you know, tracking, but they're all going to have some kind of noise. But you can combine all, you know, multiple sensors and other information to make it much better. You know, things like other sensors or models. So for example, you know, we all talk about, oh, you should put a GPS on something and measure its position. But, you know, if a GPS is just standing still, this is the kind of sensor that you would get back. So generally, good GPS would be 1 meter accurate. So, you know, that's what the sensor would output. So the other thing is reliability. So sensors can sometimes fail. So, you know, you could be passing through a tunnel, you know, and your GPS suddenly just gives you really erroneous measurements. We did a project a while ago where we stuck an IMU and a magnetometer to a cheetah, and there was a certain point where the magnetometer would just go crazy, and we think that there may be some power lines underneath there, right? So your sensors can go and do some crazy things. But if you had more measurements, you can then employ some kind of outlier rejection, right? So for example, in that figure over there is a GPS from a car. So the car is going through a tunnel, and the bottom is a radar's velocity. So you've got to fit it with a radar, and it's giving you, you know, the ground velocity of the vehicle. You can see it's unaffected by, you know, going through a tunnel. So you could maybe trust that sensor more through some kind of sensor fusion algorithm. It's also the problem of missing data, right? So the GPS, a really nice, expensive GPS, can give you 10 Hz update rate, which of course, like if you're trying to do biomechanics or any kind of, you know, kinematic measurement of an animal, it's not great. So, you know, this, you can see in the graph there, that's a simulated car turning, and you can see that, you know, it's quite patchy, the data from the GPS. So it's not going to be ideal if you're trying to do any kind of reconstruction. And then there's also unmeasured states, right? So, you know, what if your sensor, in our case, the camera, doesn't directly give you the thing you're trying to measure, right? So if you think of a camera, it's actually not giving you the, you know, the shoulder position of your animal. It's giving you a pixel, which is somehow related to a camera matrix, which is somehow related to, you know, the 3D kinematics of the animal. So as an example I always think of is if your GPS gives you, you know, position, but it doesn't give you velocity. So if we just straight up differentiated that velocity, we took the derivative, you know, it would be super noisy, as you can see in the top left there. And we could filter that, but obviously if we filter, we are removing a lot of information, you know, through something like a low pass filter or something like that. It's not ideal. So the Kalman filter is my preferred choice for sensor fusion. And sort of the I think it was invented in the 1960s, and it's one of the most common methods for fusing multiple sources of information and data. So it can provide you the optimal state estimate. So the optimal, when I talk about a state, I'm talking about, the variable quantity which describes the system. So, if you're following, a car, it will be the position. If you're, a cheetah, it could be the angle of the tail, things like that. And it gives you the optimal state, proven mathematically if you have a linear process model. So if your system behaves linearly, if your measurements, so your sensors are related linearly to your states, and if you have Gaussian noise. Okay, so if you have some kind of noise distribution, it looks like the diagram over there. Okay, so how does it work? Let's say you are walking, trying to get to your friend's house in some remote forest, and you got really patchy cell reception and no GPS, and how do you do it? Right? So you start at some known position, and then you'd walk for some time, let's say sometime in the future at K+1, you'd think that you are at a certain place. But then you manage to get cell reception suddenly, and your friend phones you and they say, no, you're actually, you're going the wrong direction. You need to go, you are in this location, not where you think you are. And so they say you're at the red dot. What you then do is you then weigh up, okay, how your friend doesn't quite know where you are, but they do know a lot more than you. So you now can recalibrate in a way and restart and say, okay, now I think that I'm actually with the triangle. You then lose cell reception again, and you walk a bit, and now at K+2 you get a call from your friend again, and they say, whoa, you're way off the mark, you're at the red dot. And then you can once again weigh up how what the information your friend is giving you versus how confident you think you are, and then come up with a triangle and so on. So then you can do that for going forward, K+3, and then we end up with a new state. So that's sort of how the Kalman filter works in a nutshell, is that you have a part of prediction. So that's where you have a model of a system that says, you should be at this state. And then you have a measurement, and the measurement says, I think you're over here. But the measurement will also have its own uncertainty. So I'm showing you the state space formula, but basically this is essentially a model that predicts, given the current state information, so the current angles or the current angular velocity, and the current input to the system. So, and if you have a robot arm, for example, you could have the torque applied to the joint, and how does that relate to in the next sample? How is your state going to change with time? And then also we know that the model isn't 100% accurate, so it has some noise associated or uncertainty associated with it. And then the other part is the measurement model. So that basically relates the measurement that you have, the camera pixels, the GPS measurement, and how is that related to your states, right? So how is it mapped into, how do your states, so your angles, your angular velocity, how do they get multiplied and added to give you, the pixels? So like Glenna mentioned, you'd have a 3D point that then goes through a camera matrix, with a camera pose in it, and then being divided by the focal length, and that ends up giving you, the actual pixels that you would measure. And then as well, measurement noise, right? Because the actual camera isn't going to be as accurate as you think. So the first step of the algorithm of the Kalman filter is that you have a prediction stage. So that says, given my current state and my current input, where will I be in the next sample? And then you also have a covariance, right? So that is a nice and neat thing that tells you and determines, given your current state and how certain you are of that state, what's your next state, and also how uncertain will it be, right? So if you haven't had a call from your friend in a long time, in the example, you would imagine that your uncertainty on your position would grow, with time. And you can see that uncertainty, or state covariance, as we call it, is determined by the model's covariance. Right? So how accurate are we, and how confident are we in our model and our prediction. And then the next step, or the final step of the Kalman filter, is an update step. And here we calculate the residual. So this is the pre-fit residual. So given our states, we can work out what our measurements should be. And then we have our actual measurements, and we subtract those, and that gives us our measurement pre-fit residual. We can then work out the predicted measurement covariance. And here you can see that's the sensor covariance. So for example, in deep lab cut, we know it's accurate, say, within 10 pixels. So that will be, the covariance of the measurement there. Then don't worry about how that's derived. I've got some links at the end there, but you then get the Kalman gain, which is basically a correction factor. You can see over there, there's a K multiplied by your residual. So given, your prediction and then your measurements, how much do you need to pull or push your state prediction to get you a new and updated state, right? So obviously, if your measurements are really good, then you'll have a high Kalman gain, because it will try and always pull you towards your measurements. And then we can also use the Kalman gain to calculate a new uncertainty or covariance. So now we've got a new state, and we've got how uncertain we are of that new state. There's a lot of math there, so I think if at least today I give you the concepts across, then I'll be very happy. Okay, so let's look at an example. So this is a car being navigated. So on the x-axis, we have the position, and on the y-axis, that's just time. It's not the 2D location of the car. So the car, it starts out at 0, and it sort of like accelerates and just keeps going. And you can see how it's moving. So what we want to do is estimate the position and velocity of the car at 1 kilohertz. So at our disposal, we have a GPS sensor, which has an uncertainty or standard deviation of noise on it of 1 meter, but it's also coming in at 10 hertz, right? And remember, we said we wanted to do it at 1 kilohertz. And then we've also got a wheel sensor, so that measures the angular rotation of the wheel, and that gives us the rotational rate of the wheel with an accuracy of 0.5 radians per second, but that's coming in at 1 kilohertz. So the other cool, neat thing about the Kalman filter is we can have sensors of multiple rates. And this is quite useful when you have cameras where, sometimes the one camera doesn't see it, but the other cameras, the other 5 see the shoulder of the cheetah or bird or whatever you're tracking, then you can actually, the Kalman filter will still work. So in this example, our states are the x position. So this is a 1D example of cars just driving in a straight line, and also the velocity, right? So the x position and the x velocity. So how do we do the update, the process model, the prediction model? We need a way of predicting how, the position and velocity change as a function of time. But we also have the wheel sensor, right? So what we could do is propose this model. And what you'll see there is that we have the next velocity and the next position are linearly related through, you can see the top differential equation over there just relates, it says the wheel's rotation, u multiplied by the wheel's radius, will then give you the velocity if the car is moving. We're assuming it's not slipping or anything funny like that. And then the position is made up of the time derivative, the integral of the derivative of position, which is velocity. And that's the second row over there, right? So using that information, and if everything was perfect, we could just predict where, the 1D location of a car purely by looking at the wheel sensor. Okay, and then we need a way to write the measurements. So the measurement in our case is the GPS sensor as a function of the state. And this one is quite easy. It's just because the GPS sensor gives you the position already, we can just put a 1 there, right? Because it's just 1 multiplied by the state gives you the position from the GPS, right? But just be careful. I remember when I first started doing Kalman filters, I always assumed that, you know, the sensor would give you the measurement, but it's not always the case. You know, for example, in the camera, like Glenna mentioned, you know, it basically converts 3D points into pixels. So that's what will go in that H matrix over there, which I'll go through an example in a bit. Okay, so now we've got the models prediction. We've got the update step. So now we need the uncertainty. So the main uncertainty on the input, is the input to the model, which is the wheel sensor noise, which you said is 0.5 radian per second. So you can see that covariance of the model part is written as sigma of the wheel, which is 0.5 multiplied by the radius, because we want it in velocity in meters per second, and it's squared because we're looking at covariance, not standard deviation. So in this, we've assumed no cross correlation and that the position model has no errors, right? In reality, there's actually a more accurate way of calculating that, which I do in the MATLAB live script. I'll talk about in a bit, but this one is pretty good as an initial step. Okay, the next thing we need to know is the measurement covariance. So this describes the uncertainty in the sensors, and as we said, the main uncertainty was you know, the 1 meter accuracy on the GPS. So R, is it actually a matrix this time? It's actually just a scalar, but if you had multiple sensors, you can imagine it becomes a matrix. And again, you know, if they're correlated, you would have terms between them, but generally you can assume that they are uncorrelated measurements, right? So if the accuracy, for example, of one camera seeing the tip of the tail of an animal is unrelated to the accuracy of, you know, seeing the second camera seeing the tip of the tail. Okay, so the next thing we need to do is initializing. So you remember, you needed to know a place from where to start in order to actually in order to actually like propagate forward, you know, where you think you're going to be. So you need to initialize your state. So your initial, in this case, your position and velocity, that's your x naught, and your state covariance. Right? So how accurate do you know your initial point, right? So if you started at a parking lot, you know, and it's really accurate, and you've got the GPS location, you know, then you could say, you know, it within 10 centimeters. So that would have a small covariance. But if you are, you know, starting just randomly, somebody dropped you in a forest, it might be tens of meters or hundreds of meters. Right? So in this example, let's just take a guess and say, okay, we know we're starting at the origin, and we know, you know, our velocity is also starting at 0, but we know that within 0.5 meters position and 0.1 meter per second covariance. So our x naught, so our initial state estimate is going to be 0, 0, and then our initial state covariance is going to be 0.1 squared, you know, because that's the velocity states covariance, how certain we are of it, and then the 0.5 squared is for the position accuracy. Okay, so how do we do this? We can then this is pretty easy to implement. If you check out the folder, you'll see that we can run the prediction step at 1 kilohertz. So all of the stuff, I'm a MATLAB fan. Okay, so all of my stuff's written in MATLAB. So you run it at 1 kilohertz, and then you can, but you'll see that we have a counter in the code that only checks when the counter gets to 100, and then you run the measurement of the GPS, right? Because it only comes in every 100th sample to simulate that. So you can see here, this is the output. So the ground truth is the blue line, the red's the GPS. You can see there it's quite, you know, far spaced in time, but also quite easy. So what happens when we initialize it? Let's say I thought I was at 5 meters instead of 0, and I knew that within 2.5 meters. You can see, so changing the covariance, you can see the filter starts off, the black line starts off quite far off, but quickly converges to the solution over there. So the Kalman filter is pretty robust in that way, provided you know how to set it up properly. Okay, I'm going to go through this quite quickly because I'm running out of time. But let's say we're trying to estimate the angle and angular velocity of a pendulum. So let's imagine you have a pendulum and it's swinging back and forth, but it's driven by a motor, right? So it can actually swing to any angle. So this is the sort of angle profile you get. It starts from the bottom and it swings all the way to the top. So our measurements that we have in this example are a rotary encoder. So that's an angle sensor and it has an accuracy of 5 degrees. But specifically, I wanted to show this example because we cannot measure the torque being applied to the pendulum. So, you know, for us as biomechanicists, you know, we will never be able to know exactly the torque being activated, the magnitude of that torque, say on an animal, right? So we need to do some tricks, and I'll show you in a bit how we do that. So again, here the state is just going to be the angle and the angular velocity. So the process model, like if we had a torque sensor, or if we could know, you know, the input to the system. So for example, if you maybe had an EMG, that might help you a little bit because you'd actually know muscle activation, but you still wouldn't know magnitudes of activation. So that wouldn't be that great. So what we can do is we can assume that the motion of the pendulum, because of the fine speed and that we are sampling, is mostly a constant acceleration within 1 millisecond, right? Because we're trying to do this for 1 kilohertz. And that's what's called a constant acceleration model. And what we then do is we are trying to estimate the velocity and the angle, but we introduce an extra state for the acceleration. And what we do over there is we say, you know, yeah, we come up with this new model, and you can see we have the acceleration, the next state's acceleration. So the next time step acceleration is just equal to the previous step's acceleration. You can see it's just equal to. And then we have the other two differential equations are just the integration of, you know, velocity, acceleration, being integrated to get velocity and integrated to get position. You can see these. So that's how we've modeled this as a constant acceleration model. And also notice there's no control input, right? So because we can't measure the torque on this pendulum or our animal that we're trying to model. So we do have an encoder. So the encoder is giving us the angle. So, you know, the measurement model looks like that. And again, I just can't stress this enough. It's not always the case that you directly measure the state. So the main uncertainty in this system is the changes in angular acceleration, right? So how often or how much can this model or this system deviate from being constant acceleration? So the rule of thumb is to choose, you know, to calculate what you think the biggest change in your acceleration will be, and then you choose somewhere between half and that maximum value. That is, if you've done a first year class in physics, is sometimes referred to as the jerk of the system. But that can be our tuning parameter. And for now, let's just set it to be 14 radians per second squared. So that's sort of what our covariance of the model looks like. And the measurement's pretty easy, you know, as long as we need to put it back into SI units. So we got our 5 degrees needs to be converted to radians per second. So initializing, again, like I said, we need to do state covariance, and we can guess. We know that the pendulum is starting straight down, and we know that, say, within 5 degrees, and we know that it's at, you know, it's not accelerating, and it has no velocity, and we also know that, say, within 0.01 accuracy. So that's where we can see those values come from. So we can simulate this, and you can see this is with you know, showing the acceleration noise set to the maximum, so the 14 radians per second. And then if we reduce that considerably, like we say that the system, you know, can't, you know, the uncertainty on the change in acceleration is much lower, so we reduce that quite dramatically. You can see that the state estimate gets way smoother, right? So you can see it gets much better looking. Okay, very quickly, Kalman filter only works for linear systems, but for all of the stuff that we care about, actually in 3D kinematics, it's all actually nonlinear. I won't get into it, but follows the same formula. You just need to linearize your model or your measurement equations over there using a Jacobian. Don't worry, I've linked some stuff at the end where you can see how to do that. And then once you have those two matrices, you do everything like you did before in the Kalman filter. Then also for observing animals and doing sort of biomechanics analysis, we often can do this stuff offline. Right? So you don't have to do it, at a sample by sample basis. We actually maybe have like a full video that we're trying to analyze. So we can actually use information about the full sensor, collection. And that's what's called smoothing. And again, I'm not going to get into the maths of this, but essentially how it works is you run a Kalman filter on the forward pass, and then you run backwards. So now that you have state estimates and estimates of your covariance, you can then run backwards in time to get a much better estimate of your state, and that's called the smooth state. And they follow those equations, and you'll see in my code how to implement. Okay, so skeletal models are what we need when we're trying to do 3D reconstruction, and skeletal models add information about the animal's physiology, so that what it looks like. And then we also can add temporal models, right? So how it changes with time. So that one example of temporal model is the constant acceleration I showed you. Okay, I'm going to try and go through this very quickly. I assume most of you have done some kind of math first year math or some kind of thing like that. But if we have a point in one frame, in your world frame, like Glenna mentioned, and you want to express that in a new frame, one over there, we can relate those if they, for example, are rotated by an angle psi over there. We can relate them through a rotation matrix, right? And the notation I use is, the one at the bottom represents a rotation that takes something from frame 1 into frame 0, which is the superscript. So we can take a vector in 1 and produce what that vector would be in frame 0. Okay, and in that particular case, it would be over there. It would be sort of an elementary rotation about the z-axis, would be something like that. And you can work that out. You can derive it, with trigonometry, but that's sort of what you'd get. And then we can also relate the other way around, right? So we can relate something in frame 0 to frame 1 using the rotation matrix the other way around. So then you have these are fundamental rotations. Again, these are sort of, you can get them on Google, but any 3D orientation of an object, animal, robot, can be expressed by some combination of these 3 fundamental rotations. Rotation about X, Y, and Z. So if you want to change axes, we need the inverse of the rotation matrix, and there's a nice property that rotation matrices are orthogonal, so you can just calculate the transpose and it'll give you the inverse. Here's just a quick example. Let's say the position of a nose in an aircraft frame. So let's say that the nose is 20 meters out in the aircraft's x-axis and 0 in its y-axis. So how do we calculate the nose of the position in the inertial frame? So we can use the rotation matrix, because it's just a rotation matrix about the z-axis with an angle of alpha. And what if the aircraft started moving? We can calculate the nose with respect to the origin of the world frame using something like that. So you can see that we first need to rotate the position of the nose in the aircraft frame, rotate that back into the world frame, and then you can add that to the position of B, which is the motion in the world frame of the center of mass. I hope that makes sense. And then we can apply successive rotations to get any arbitrary 3D orientation of an object using multiplication of these rotation matrices, and the resultant matrix is called the direction cosine matrix. The book that Glenna mentioned, is Hartley and Zisserman, is a really good one. You should check out. And then we also have these sort of easy ways of thinking of rotations called Euler angles. So for example, and these are 3 numbers which represent the orientation of an object. So for example, Euler 3-2-1, you will hear people talk about, is a rotation first about the third axis, which is the z-axis, then the rotation about the second axis, which is the y-axis, and then the rotation about the x-axis, which is the first axis. There is a problem, and that's called gimbal lock. So you can check that out. There's some cool animations online, but basically what it means is when the second rotation in your pair, so for example, when the y rotation goes to 90 degrees, you get an undefined solution. But so that you have to be quite careful when you're defining your Euler angle rotation sequence. So for example, for us tracking cheetahs, we know that the cheetah can't roll past 90 most of the time in the videos that we look at. So we use that as our second rotation and avoid the singularity. Okay, I've got 15 minutes, so I'm going to try and show you an example. So my group is quite interested in trying to understand the head stabilization of the cheetah. So you can see, you know, despite it's running really at high speed, the head is quite remarkably stable. So I'm going to show you an example of how we can track the motion of the head using deep lab cut. Okay, so I'm going to jump to. Oops, I'm going to jump to my MATLAB. Can you guys see my MATLAB window?
Yes, we can see it. Yes, we can see it.
Okay, awesome. Thanks, Brooke. Okay, so I'm going to go through this very quickly. All of this is up on the drive. So we're trying to model the head and the neck of the cheetah. So you can see the head is the pink triangle over there, and it's defined, you know, using deep lab cut. Deep lab cut will give us the eye positions and then also the nose. You can see over there. So those are the circles. And then deep lab cut also gives us the neck, so the base of the neck over there. And we can define them as, so the head, imagine the axis of my head, can move in 3 dimensions. It can translate, so it has an X, Y, and a Z over there. That's the head written over there. It can also rotate in 3D. So it has 3 angles that define its rotation. So there's 6 degrees of freedom with respect to the world frame. The neck can extend, so that L over there can actually, you know, lengthen and contract. And then the neck itself can rotate about the y-axis and about the z-axis with respect to the head. So that's a relative rotation. Okay, so we have 1, 2, 3, 4, 5, 6, 7, 8, 9. Sorry, L is 9, the 9th state. So we have 9 states. So 9 quantities which represent the system. And those are the ones which we're going to try and estimate using our cameras. Okay, I hope that makes sense. So I'm just going to walk you through this code quite quickly. So we define some symbolic variables over here. Can run the section. Okay, so that just puts them in the workspace. Now we're going to use a constant acceleration model here again. So for every, you know, quantity or degree of freedom, like I mentioned, it needs a velocity and an acceleration state. So remember I said we have 9, you know, degrees of freedom. So we're actually going to need 9 times 3 states. So we need 27 states. So you can see I've written them out there as position states, velocity, and acceleration. So our full state vector looks something like this. So you can see as ddx, ddy, ddz, all the way down to L, you know. So here you can see all of the states over there. Okay, and then we define the rotation matrices. So like I said, I know that the cheetah doesn't roll its head past 90 most of the time. So we define that as our second rotation. So we rotate about Y, then X, then Z, and that's defined by these angles. And that represents a rotation from the inertial or the world frame into the first frame, which is the head frame. And then we can also define the rotation from the head back into the inertial, and that is just a transpose of the previous matrix. And then we can do the same thing for the head over there. So let's run that section. You can see there's our rotation matrices written out quite nicely. Okay, the next thing is we then need to define a way to get our positions of our states and where they are. Sorry, the position of our markers in terms of our states. So we can write these out. These are just kinematic equations over here. So you can see, like I said, if you want to get the position of something in the world frame, you need to add, the previous vector. We need to rotate it into the world frame, and then we take the vector defined in the head, for example. And yeah, you sort of get the idea. You can do that for the eye, the nose, and the neck base. We run that. So you can see these are getting quite hairy right now. Okay, so now the next part is the prediction part. So like I said, we have a constant acceleration. So we can say the prediction equation for the acceleration is just equal to the acceleration of the previous step. The prediction for the velocity is the integral. So this is just the first order Euler integration. We then have the position, which is then just a second order integration of that. And then we lump all of those together, and those are our equations over there. We also have a matrix. Whoops, let's go and go down here. Okay, so there you can see. So yeah, these are all the equations. You can see, and it also makes it quite neat. And then you can also, this is just the F matrix that's needed for the Kalman gain. Okay, next. Now we need to define the measurements. So I should probably do this. So so my students like Python. I don't, but so all our stuff, our cheetah work is done in Python. I'm going to link something at the end. So this is just something that I wrote, which takes the JSON file, which is Python's data format, and converts that into MATLAB struct for the calibration. So once you've done the stuff that Glenna has spoken about, you can get the rotation matrices, the translation of the camera, the distortion parameters, and the camera matrix, so the focal length, and it's the center of the image plane, things like that. So now we've got all the camera stuff. There's a function I wrote, which is also up online, which takes the position of the eye, so the 3D position of the eye, these camera parameters, and then produces what the predicted pixels would be. So I'm gonna show you what that looks like. Yeah, there's a bit of time. So this basically is a fisheye camera model, because we're using GoPros and they have horrible fisheye. So it first takes the position and the orientation and translation of the camera, and converts that using a pinhole model to get the pixels, and then applies the distortion parameters. And these are well documented in that Zisserman book. Okay, cool. So then we can run that guy, and then we end up with all of our equations. And there's also some other functions at the bottom here. Okay, so now we have our equations that we need. So let's run the Kalman filter. Okay, there's a couple of things to look out for. So you need to define a beginning and an end frame. So you usually want, this is tricky because when you're dealing with things in the wild, because you're not going to see the animal all the time. So it really cuts down the amount of data that you can extract and analyze. So you need to find ones where at least two cameras are seeing the thing you're looking at. So in our particular case, we only have about 80 frames, from frame 2 to frame 80, where we can actually do 3D reconstruction. Okay, so another thing to note here is you need to define the accuracy of the DLC measurements. So like I said, they're not perfect, and that you can get from when you're doing training of DLC. As Glenna mentioned, there's lots of information on how to do deep lab cut model training, but that's sort of something that you would spit out. So deep lab cut also gives you a likelihood of a marker. So, it will tell you if it's not very certain. So I like to set a high threshold on that of 0.8. So 1 is where it's like completely certain, 0 is where it's saying don't trust me. So anything above 0.8 is a valid measurement. Anything below is discarded. Okay, let's see. Let me run this quickly. Actually, let me just run all of it. Okay. Okay, it's doing its thing over there. So these are the parameters. So the lengths. So you need to know the geometric lengths of the animal. So with this kind of kinematic model, and you can get those by manually, getting a grad student to to click the eye positions for you, and then do some like sparse bundle adjustment triangulation and get the metric distance. You can try that on multiple animals. So we've done this for you already, and we've got the parameters for the cheetah over here. So then we load in the deep lab cut labels. So there's a link there on how to convert the deep lab cut output into a CSV, and this will read the CSV for you. This is a bit annoying. I wish there was a better way to do this, but you basically need to look at the columns and determine. So for example, in my case, we're looking at the right eye and left eye. So that's column 1 to 6. And then we need the nose and the neck, and that's the ones at the end. So that's the from number 5 to the end. And what this does, it just calculates where the pixels are that give me a vector, and where the indices of the likelihoods are. So that's likelihood index. Okay, next is we do initialization. Let me see if I can zoom out a bit. Yeah, it's a bit better. So we need to initialize all the states. So we initialize the acceleration to this 0. We then initialize the velocities as well. So the values here have been hand chosen and tuned, so they are the 100% correct ones. So I just multiplied by them by 1.5 just to show you, that the filter would still work with an okay initial state, and then the same with the position. We then have to give the initial state covariance. So here again, for example, the position of, in this case, the cheetah's head is about 2 meters away from the world frame, which is defined at camera 1. You can kind of eyeball, like it looks like it's going at 5 meters per second, all those kinds of things, and you put down that uncertainty, you quantify that uncertainty over here. You then define the process noise. So I've done a bit more of a complicated process noise and the uncorrelated one that I showed in the slides. And I've linked something here where they actually derive it, but it's written out over there for a constant acceleration model. And then this part is we actually run the filter. So again, this is just straight out of the slides, but I just wanted to show you, so you can see here, this is where we do the prediction. So here at this point, we calculate basically for the low likelihood points of deep lab cut, we just increase the noise on them. So we say we really don't trust them, so the filter basically discards them. This calculates the residual, so the prediction from our prediction step minus the ZOK is the pixels, which is the actual measurements that we get from deep lab cut. Okay, and this is quite an interesting thing we had to implement. So you can check out the link here, but we had to do some outlier rejection for the Kalman filter. So sometimes deep lab cut will say with a very high confidence that it's found a point, but that point is way off. So how it works is it basically checks the predicted state sensor covariance. So let's say the state covariance should be 30 pixels, and it calculates the residual. And if the residual is more than 3 times that, I've chosen 3, it's because that's sort of the customary thing to do. It's called the 3 sigma method. You then treat that as an outlier, and you don't update using that measurement. So that's what this little bit of code does over here. We then calculate the Kalman gain and the covariances and so on. We then do the smoother, so a backwards pass. And you can see on the right here, this is that's the prediction from the smoother. It looks way better than the actual Kalman filter. And then just for fun, I showed this is an animation. So that's the cheetah's head and neck base, sort of running past the camera. Okay, I'm going to go back to the slides quickly, sort of finish up. Okay, so the Kalman filters are great, but like I said, they require really good initialization. So I mean, I've kind of like tried to make the initialization worse, but they generally, you play around with that filter enough, it will not converge and go unstable. It can't handle constraints. Okay, so for example, your spine in the cheetah's example can't bend, upwards like that. It can only bend like downwards like that. So you can't add that into the Kalman filter. So sometimes it might give you solutions with the cheetah's, bending its limbs or its spine or its tail in really unnatural positions. So it's much better to do what we call a full trajectory optimization or full trajectory estimation, which is a paper we wrote last year. And this is a very quick punt of that. You can see this is the full cheetah running there, looking pretty much like a Vicon to me. And that's the comparison in the paper we do on 3 of the methods: the Kalman filter, the SPA, which Glenna mentioned, and then the trajectory estimation. Okay, so all of that code is up online. It's on GitHub for a Sino set. The full data set of our 3 years worth of cheetah videos is up there as well, and you're welcome to play around with it. But yeah, so for further reading, check out Brian Douglas's videos on sensor fusion. There's a really nice Jupyter notebook course on GitHub, which really does the fundamentals of Kalman filters. And then also check out our paper, Sino set, which is our full cheetah data set, as well as a lot of this code in Python though. But I think most of you are probably comfortable with that, and how to do reconstruction using full trajectory estimation, which is a much better way of doing it. So yeah, thank you very much. I don't know, there are like 1 or 2 questions we could take now. Are there any specific MATLAB toolboxes we need to have installed to run my code? No, you should be able to run it on standard MATLAB. I'm sorry. No, you need symbolic toolbox.
Great, thanks.
Yeah, but if you don't want to rederive the equations, you can just run the Kalman filter script, and you don't need it.
Okay, thank you, Amy. So
Cool.
I can go next.
Yep, let me stop sharing. Thanks.
Okay, so now moving from a single individual, my presentation would cover on topic related to when we have multiple individuals that demonstrate collective behaviors. Specifically, I'll talk about modeling and data analysis techniques that we can use to study such collective behaviors. Let me start my laser. Okay. So let's start with some collective behavior videos. Those are always fascinating to watch. So what we are looking at is a video of like starling flying together, and you can see all they're making like beautiful patterns, but there are no leaders who are in charge of these beautiful patterns. It's just like each individual is interacting with a subset of the group, and these interactions are giving rise to a group level behavior that we are observing. Outside starling, we can also see such behavior in fish schools, where you see they're swimming together in the same direction. So today I'll take an example of a very different system, which is going to be our motivating example. And that would be a system of bats, because they are special, and we'll talk about it, why they are special and the way they communicate. Next, I'm going to talk about like how we can use different data analytic tools to learn about these real world interactions, and a simple model that we can use to study such collective behaviors, and how we can quantify the group level behaviors. Finally, I'm going to take this simple model and modify it to represent some of the bat sensing mechanisms. And finally, I will conclude with showing that how these IT tools, I'm referring information theory as IT tool to distinguish the sensing modes. So with the last couple of talks, we saw like the importance of tracking and also the importance of data. So you will see the other side, like what we can do when we have the data. So now let's get started with the first example here with the bats. So why bats? Bats are special. They execute collective behavior, but unique about bats is that they use echolocation or active sensing. That means they generate their own ultrasonic sound. The sound gets reflected from the obstacles. They listen to that, and that's how they detect the obstacles, and that's how they navigate. An important component of echolocation or active sensing is it promotes information sharing. There has been some biological evidence where it shows that when two bats fly together, oftentimes one bat just may stay silent and eavesdrop and listen to its peers' echolocation and can detect the food source or the obstacles. So this is like how bats effectively use active sensing is of interest to the biologists as well as engineers like me, because we can learn from this robust system and design engineered system. For example, energy efficient drone swarms. So to do that, what we did is we focused on a pair of bats to understand how they communicate. And here you can see this is our experimental site. This is what I pursued during my PhD studies. Here, the two bats flying out of the cave. One is in the green, that's the front bat, and the one in the rear end is marked in yellow. So we had 6 different camera views, what Liana mentioned, like how the different ways we can track. So I was mostly the data side, but our research group went to China. This is our experimental site in China. And we had 6 different cameras set up, and the bats are flying out of these caves, and we tracked the pairs of bats. So here the research question is, when 2 bats fly together, do they communicate with each other or not? And in case they do, what is the directionality? Is the front bat influences the rear bat, or the directional influence is in the opposite direction? So to answer the research question, here are the steps. So first, collecting data, then tracking what we saw today earlier, how we can use different camera setup and track the 3D coordinates. Here we tracked a total of 30 pairs of bats, means their XYZ coordinates, their trajectory point. And once we have that trajectory data, we converted this to 1D time series data in form of curvature and used appropriate data analytic techniques to answer our research question. So since it's a motivating example, I'm gonna go over these tools later, but let me share you what we found. So we used 2 tools, 2 data analytic tools, and both those tools unanimously detected that there is a significant evidence of rear bat influencing the front bat. But this is counterintuitive to us because we humans use passive sensing. We have to rely on vision, and we cannot process information from our rear end because that's our blind spot. But for the bats, they may be using this audition-based sensing, where you can think of the rear bat making a sound, the sound propagating forward in space. The front bat listens to that and may respond. So because of the sensing mechanism they use, that may provide this rear to front coupling that we're observing over here. So at this point, I would like to just make 2 comments. First of all, it's important to realize the importance of data analytic tools that can allow us to learn about these directional relationships, so we can learn about the nature of interactions. And also the second comment is like it allows us to ask more questions, how these sensing mechanisms lead to different, or whether it leads to different group level behavior compared to when we use like vision-based sensing. So to answer that, we need to rely on modeling techniques. So today I'm going to try to give you a flavor of the data analytic tools that we can use, and also the simulation that we can conduct and modify them based on what we are trying to answer. With that, let's get started with the second component, data analytic tools. So what we are doing with these data analytic tools. So let's say we have 2 time series data, one in red marked as X and Y. You can think of these 2 time series data corresponds to, let me open the chat option. Okay. Yeah, so let's say we have these 2 time series data from 2 bats, and they are representing its curvature. So high value of curvature means the bat is taking a very sharp turn. So once we have these 2 time series data, the question we are trying to answer here is how these 2 time series variables are related. So if they are related, there are 3 possibilities. For example, it may happen that X influencing Y, or it could be Y influencing X. These are 2 instances where we have unidirectional influence, or the third possibility, we have a bidirectional influence, means both X and Y has significant influence on each other. So how we can extract the relationships that the time series variable share. To do that, what we rely on is causal discovery tools. One such tool uses information theory, transfer entropy. That's what I use in my back study. The second one I used is called convergent cross mapping, which is based on dynamical systems theory. So these two tools I have used earlier. As part of my research, I test these tools that are developed by other research groups, and as well as we develop our own tools for this same purpose of detecting the causal variable from time series data and apply to different collective behavior studies. So today we're going to focus on the information theoretic tool only. So just to understand how this tool works, I think it's best to get started a short topic on what is information and how we can measure information. So to get started, what is information? Let's conduct an experiment. Let's take an intuitive understanding what is information. So let's consider an experiment where we have a biased coin, means we have both sides head. And what is the outcome of this experiment? Is there any surprise? So there is no surprise, because even before the experiment has been conducted, we know what the outcome is. It's always going to be a head. Now, compared to if I take the second experiment where we have a fair coin, means there are possibilities of either a head or tail, which are equally likely. And now if I ask the same question, what is the surprise in this experiment? So this one seems like, compared to the last one, has a surprise because we don't know the outcome yet. So the surprise is whether it's going to be a head or a tail. The result of which experiment has more uncertainty? Of course, the second one, because before the experiment was conducted, we didn't know the outcome. Once we have the result, we see what the outcome is, we are learning a new knowledge. So that new knowledge is reducing the uncertainty that we had previously. So this is what is called information. So information is directly related to uncertainty. It reduces the amount of uncertainty. Any knowledge that we gain that reduces the uncertainty is basically information. So that probably helps you to understand what is information and how. The next question is how we can measure it. So to do that, we have to refer to the seminal work by Claude Shannon in his landmark paper, A Mathematical Theory of Communication. He came up with a way to measure information. He's also known as the father of information theory. And he introduced what is called Shannon information content. So the Shannon information content for an outcome with a probability P is defined as log base 2 1 over P, which can be also written as minus log 2 of P. A question may arise like why we are using log function over here. There are several reasons, but a couple of them are very intuitive as well. So we just learned that when there is a deterministic outcome, there is no information. So there is no uncertainty, means there is no information associated with it. So log function satisfies that property. When we have P equals to 1, that means there is no information. Secondly, when we have more surprising events, means that are less likely, that would have more information again associated with it. Just to give you an example, I'm currently in Florida. If I come to know it's going to be snow tomorrow in Florida in summer, that's going to be a very surprising event. So that will have a lot of information associated with it. And that's what we want. And again, log function satisfies this property that information content increases as we decrease the probability. So it's a physical quantity. It has an unit associated with it. So it can be either bits, means binary digits if you're using log base 2, or it could be natural units when you're using log base E. So going back to the example that we started with, tossing a fair coin, which had two outcomes, heads and tails, and the Shannon information content corresponding to each outcome is 1 bit. So that helps us to understand how we can now quantify information. So what we learned so far. Okay, so let's do one quick activity just so that we can get a good hold of how to compute information. So we'll play a game here. So we have, let's say, there are 16 boxes, right? And exactly one of them has a chocolate in it. And assume that I don't know what the chocolate is. I prepared the presentation, so I know. But assuming that we don't know, I'm going to try to open each box at a time and see whether it has a chocolate. And at the same time, I'm going to measure how much information I'm gaining. So let's start with the first attempt, and I missed in the first attempt. So initially, in my first attempt, I could have missed in 15 ways out of 16 because 15 boxes do not have a chocolate. And if I plug it over here, this is the amount of information I learned, knowing that there is no chocolate in this box. Let's make a second attempt. Again, I miss. Now there are 14 boxes that do not have a chocolate, and the probability of missing is 14 over 15 in my second attempt. So this is the amount of information I just gained. Let's try one more time. Again, I missed. Fourth attempt, I missed. Not a lucky day for me, but this is a trend over here that we can see. As I'm missing more and more boxes, this is the information we are gaining, and there's a pattern that the information is increasing. Why is this? Can anyone tell me? Okay, I'll try to give you an answer here. So as we are opening more boxes, the chances of missing in my next attempt is gradually decreasing. So we talked about that as we have probability decreasing, the information content would increase. So we are seeing over here there's an increase in the Shannon information content as we are opening more boxes, and we know that those boxes do not have a chocolate. So let's make another attempt, and this time I found a box with the chocolate. And this is the amount of information I gained when I learned that this box has a chocolate in it. So now if we add all them together, we get 4 bits of information. So this is the amount of information we need to identify the box with the chocolate in it. A question may arise, what if we start the game in a different seed where the chocolate is located in a different box and we take different number of attempts to find it? So let's see. This time, I don't know. Okay, this time I'm lucky. Found the chocolate in the first attempt. So what's the information we just gained? So there are 1 over 16 in the probability to be lucky in my first attempt. And we again, plugging it over here, we just gained 4 bits of information. So this particular game requires 4 bits of information to identify the chocolate, the box with the chocolate. So hopefully this activity and example helped you to realize few things. First of all, uncertainty and information are related concepts. Surprising events have more information associated with it. And we also learned how we can measure information, which is the Shannon information content. So now we have the background. Now we can jump on what is called transfer entropy. So transfer entropy is basically an information theoretic concept, and it's an extension of Shannon entropy. We just learned about the Shannon entropy. It measures the uncertainty associated with a random variable with a density PDF denoted as px. So this is the definition. Now, what transfer entropy does, it quantifies the information flow between 2 time series variables. And here is how it's defined. So transfer entropy from y to x measures how much y influences x. And here is the definition. I would like to draw your attention in the numerator and the denominator term. So these are the conditional probabilities, and subscript over here denotes the time index. So what we are looking at in the numerator is how much the current state of y can predict about the future outcome of x. If y does not add any additional information based on what we already know about the prediction of x from the current state of x, you would notice that the numerator and the denominator is going to be exactly the same. So these 2 conditional probabilities are same if y does not add any new prediction about x. So log of 1 goes to 0. Transfer entropy from y to x is 0. That means y does not influence x. So when we have 2 time series variables, we can compute these 2 quantities from y to x and x to y. Generally, those are 2 asymmetric quantities, and we can compare this to find the dominant direction of coupling. So this is one of the tools that I have used in my earlier studies with bats. So a question may arise, okay, so when we have a pair of bats, this makes sense to use a pairwise transfer entropy to identify what is the directional relationship. What about if we have 3 bats? Should we use this pairwise transfer entropy? Probably no, because we have to be careful when we have more than 2 interacting units. So because we may have situations like where we would have a common driver effect. So this is called a common driver effect, where let's say we have a third time series process Z, which is influencing both X and Y. So in such a scenario, if I implement the pairwise transfer entropy that I introduced in the last slide, we would see a false coupling because there is a shared information coming from Z. Similarly, we can have what is called a cascade effect. This is because when we have, let's say, Y influencing Z, and then Z influences X. Again, a pairwise transfer entropy would show a false coupling between X and Y. To avoid that, we need an additional conditional additional conditioning, which is called a conditional transfer entropy, also called causation entropy. So that would be basically conditioning on Z to see how much is the direct influence of Y on X and vice versa. So we have to be careful over here when we have the research based on the research question, and also how to use the tool effectively. So question is how we can implement this information theoretic tools. So there are different softwares now currently exist that are developed by different research groups. The one that I have been using is called JIDT, Java Information Dynamics Toolkit. And it is basically developed by Professor Joseph Lizier. And here is the GitHub link to access this. It has all the details of how to download that and install this. It's like just you have to download and you can start implementing transfer entropy or the other metrics. To learn more about this, I would also like to share a good resource that's also available in the GitHub link. So there is a series of course by Dr. Joseph Lizier. There are short series of lectures which introduces to this information theoretic concepts as well as the right way to use this software. So today, as part of the demonstration, I'm going to show you how to use this JIDT toolkit with one example. But that I will come back later. But let's see what other application of these tools have been. So information theoretic tools have been have found like a lot of different applications. Not only bat swarm that I started with, it also has been applied to fish schools to identify the leadership in fishes. It has also found like outside animal groups in neuroscience to identify basically the map, the brain connectivity map. So how the different regions of brain influence. I'm also implementing this information theory concepts to traffic and transportation, which is my other research. And also these tools have found applications even in policy making. So you can all be creative. This is an era of data, and I believe this tool will find a lot more diverse applications. Okay, so now that we got a flavor of what we can do and how we can use the tool, let's jump on the model. So I'm going to present a very simple model that we can use to study collective behaviors. Okay, so this is what is called a Viksek model. This is named after the first author of this landmark paper in 1995, Thomas Viksek. And this is a simple model, but is powerful enough to capture or replicate the flocking behavior that we see in nature. So it's an agent-based model. What it means that there are participants or particles over here that we would like to simulate. So you consider a square arena where the particle is going to move around, and these agents are given a rule. This rule is same for all agent for all time, but we would like to see how these simple rules can lead to some interesting group level behaviors or not. And this Viksek model is a discrete time protocol. So the updates happen in discrete time. There are a few assumptions. For example, all agents, let's say we have n agents that we would like to simulate. We're gonna assume that they have constant speed denoted as V naught. The second assumption is the periodic boundary condition. What it means is that, let's say, if a particle leaves through one edge of the square arena, it's going to re-enter through the other edge so that we do not lose the particles from the simulation, as well as to eliminate the effect of any boundaries in the simulation. So it's as if the particles are moving in a torus. So notations that I'm going to use is X for position. So it's going to be a two-dimensional vector because we are still considering the movement of particles in a 2D arena. The subscript would be corresponding to agent J, and T means time T. So position of agent J at time t, and Vj would be the unit velocity of agent J at time t. Note that we have already defined the constant speed. So what the particles can modify during this update protocol is their heading directions, which would be represented in terms of their unit velocity vector. Okay, so now let me introduce how this update protocol works. First, starting with the position update. It's a very simple model. So let's say for the agent J, the next update at time t plus 1 would depend on two quantities. First, where it's currently located, xj at time t, plus velocity times delta t. So v naught is the speed, vj is the unit velocity vector, and delta t is the discrete time, we choose it 1. And since it's a unit velocity vector, its norm is 1. And since it's a unit velocity vector, we can also express it in terms of the heading direction. So let's say this is the theta that the agent J is currently making with respect to the I-axis. This is my coordinate system. I can write it as cosine theta along I and sine theta along J. So this would be its heading direction. So this is how the position would be updated for all agent for all time. Now, the interesting part, how the velocities are updated. So velocity update is interesting because that's where the interactions occur. So interactions occur based on how individuals interact with neighbors. So who are neighbors? So let me define neighbors over here. So let's consider this red particle over here, which is our agent J. We're going to draw a circle around it of radius r. So all the particles that reside within this circular region are its neighbors. So this circular region is similar to its sensing region, within which it can sense the presence of particles, and beyond which it cannot sense the presence of particles. So these gray particles it cannot sense. So they are not its neighbors. Only the green particles are its neighbors. It's similar to field of vision, but it's like in a circular region. So here is how the neighbors are defined. Next, each particle, let's say here in the agent J, interacts with these neighbors to update its heading direction. So here is how the direction update happens. The direction update for agent J at time t plus 1 depends on two quantities. This is the one that's highlighted over here, is a resultant vector of its neighbors. So it learns from its neighbor what is their current heading direction, and it computes the resultant vector, and this is the heading direction of the resultant vector at the current time. And the second term that you are seeing over here, that's an error term. That's kind of a measurement noise or its own choice. So measurement noise is modeled as a random variable from a uniform distribution of minus eta over 2 to eta over 2. Eta means it's a noise intensity. So higher values of eta means there would be larger measurement noise in the system. So this is how it's going to interact, and it's going to update its heading direction. So once we know theta j for agent J at time t plus 1, I can go back and update its unit velocity vector by plugging in the definition over here, and it has its velocity for the next time step. So as you can tell, it's a very simple model. It's a very ideal model, but we will soon see it is powerful. So let's summarize how the protocol works. So we start by defining a square arena, and we randomly position all these agents randomly from a uniform distribution, and also prescribe a random heading directions. Next time step, each agent identifies who are their neighbors. It updates its position following the position update protocol, and updates its heading direction based on interaction with its neighbor, and then it computes its velocity. And this process repeats for all agents and for all time. So now let me show you what the simulation looks like. So here is an example simulation. I'm taking this from the YouTube link over here. We're going to show our simulation later. But here, just to give you a quick idea how the simulation looks like. So here, eta is the noise intensity. And here is it's the tuning parameter. So if we start with a small value of eta, means there's less amount of measurement noise in the system, we see a nice flocking behaviors. But as we keep on increasing the value of eta, that means there's more and more measurement noise, we see a random behavior. It's a random walk. The particles move in a random direction. So here is an example with eta equals to 0.1, then eta equals to 0.5. And as we keep on increasing eta, there is more of disordered behavior. So eta is a tuning parameter over here. And as we increase eta, the system moves from ordered to a disordered state. Okay, so that's why it's a very powerful tool. It's simple. We can test different model parameters and see how that influences the group level behaviors. Now let me talk about how we can measure the group level behaviors. So group level behaviors, to measure those, we need to rely on some order parameters. First of all, one common order parameter is polarization, which measures the average heading, so the group's heading. This is how it's defined. So it's basically we are adding all the velocities of all the agents at a given time and taking the norm. So assume that all the agents are heading in the same direction. So their norm would be a value n. So I'm then scaling it by 1 over n. So it can have a polarization would be having a value 1. So polarization can have value between 0 and 1. 0 means random walk. 1 means the group is perfectly aligned. So when we have the random walk, so this vectorial addition of this individual velocity vectors would cancel out, and we'll see the polarization value close to 0. So this is polarization, again, very intuitive. Next is the cohesion, to see how closely packed the group is. There are different definitions of cohesion. This is one common definition that I have used in my study. First of all, let's say this is the Ri of T, which measures the relative position with respect to the center of mass of the system. So if we think of a state where all the agents are collectively located at the center of mass, then we have Ri equals to 0. So e to the power 0 is 1. Add them for n agents, we'll have a value n. Canceling with 1 over n, we have cohesion value 1. So when all the particles are at the center of mass, we will have a cohesion value 1. And when they are spread out, depending upon, like, since this is how this is the exponential graph, and if we increase Ri, we're going to there is a negative sign over here. So as we increase minus Ri, the cohesion value would decrease and go to near 0. So 0 means they are spread out. 1 means they are perfectly at the center of mass. So these are the two parameters, the order parameters we're going to use to measure the group level behaviors. Now we have all the preliminaries. Let me just give you some examples of polarizations and cohesion. This is an instance where we have high polarization and high cohesion because, as you can tell, the group is perfectly aligned. They're also very close to each other. This would be an instance with high polarization but low cohesion because they are more spread out. This one is with low polarization because they are heading in the random direction, but cohesion is high compared to this one because they are more close to each other. And this is an instance where we have both polarization and cohesion very small values. So hopefully we have now, it's clear how we can measure the group level behaviors. Now it's time to go back and think of our motivating example of bats and how we can take this simple model and see what happens if we implement a bats-inspired sensing mechanism. To do that, what we're going to do is we'll conduct a contrasting simulation. One is where we're going to use a vision-based sensing, and the second one where we're going to use an auditory-based sensing. So let me start with a visual sensing, which is the vision-based sensing over here. Here, each particle are provided with a sensing space similar to a field of vision, which are marked in yellow. So you can think of this orange particle has its field of vision, which is this one, and the blue particle resides within its field of vision. So the orange particle can only see the blue particle and cannot see the gray particles. So in this instant, the orange particle will have only one neighbor, that it's blue guy over here. Whereas in the auditory mode, now their vision is replaced with audition-based sensing. So here they are making sound, and this is their acoustic beams are denoted by these blue regions. Now for this example, let's talk about this orange particle. This is residing within the acoustic coverage of these two blue particles. So it can hear from these two blue particles and cannot hear from the gray one over here. So in this instance, with this auditory sensing scheme, the orange particle can only listen to these two blue particles and cannot hear from the gray particle. So in a nutshell, these two distinct sensing modalities differentiates how the neighbors would be determined for each agent. For visual sensing, neighbors are the agents who are occupying the field of vision, as for example, the blue particle over here. In the auditory sensing, neighbors would be those agents whose beams are occupied. So now I'm going to run contrasting simulations, but I have to have a fair comparison. So I'm going to keep every other parameter exactly the same, even their initial conditions. And one is where they're going to use a visual sensing mode, and the second one is where they're going to use an auditory sensing mode. So let me show you the simulations. Okay, one thing I missed. So here P is the sensing angle, and this is basically half of the sensing angle rather. So if we keep on increasing P up to value pi, that means it's going to be a circle, which boils down to the original Vic-Shek model where it had a circular sensing region. Okay, now it's time to watch the simulation. So can anyone guess which one is which? One of them would be vision-based, another would be the audition-based. Again, you can participate by either raising your hand. I'm trying to open the participants window here. Let me start the simulation one more time. No? Okay, let me answer. So here, the left one is what we are observing is where the system is using an auditory mode of sensing, and on the right is the visual mode of sensing. So here we see there is completely different group level behavior that emerges based on their sensing mechanisms. So in terms of the ordered parameters that we talked about, so here we would see high value of polarization because the entire group collectively attains a common heading direction, whereas they are more spread out. So that reduces their overall cohesion of the group. Whereas in the visual mode, we see formation of clusters. So here, individuals form cluster, closely packed clusters. These individual clusters have a common heading direction, but these clusters can move around in different directions. So that reduces the value of polarization. They are forming clusters, which increases cohesion, but the individual clusters are having a different heading direction. That is reducing the value of polarization over here. Now we tested this for different choice of parameters. So let me give you some idea over here. And we use this the speed, the constant speed that we have for the Vic-Shek model 0.03. We use 1,000 particles. The sensing angle is one of the parameters, and noise intensity that we saw earlier, which is what if we increase, we see ordered to disordered state. So we want to also see how the behavior looks as we play with these two parameters. So again, like we keep the initial conditions between these two contrasting simulations exactly the same, and we ran it for 50,000 time steps, excluding the transient. And then we have the 10,000 data on which we computed the mean polarization and the mean cohesion. So I think that's where the next plots would be. Yes. This is where you can see the value for the mean polarization and for the auditory, and here is for the visual. So when we have P equals to 0, that means there is no sensing at all. There's no sensing region. So all of them has a random walk, which is evident from this polarization value close to 0. So along the x-axis, I'm increasing the sensing angle 0 to pi. Pi means a complete circle, and eta is the noise intensity, which is increased in the downward direction. The interesting thing happens when we have small values for the sensing angle P, where we see the difference between the visual and the auditory mode. And again, we see higher value of polarization as in the visual mode because they form clusters. The clusters, right. So they form clusters. The clusters are, I think sorry, yeah, this is the yellow region. So this is the higher value of polarization. So this is close to 1, whereas this is not close to 1. This is around 0.8. This makes sense. So yeah, auditory has the higher value of polarization over here because all the group has a common heading direction. Whereas in the visual mode, we see they are forming clusters, the clusters having different heading direction, which reduces the polarization. Now moving to, these are all I already covered. going to cohesion, we see again the interesting thing happens when we have small sensing angles. For the small sensing angles, we see now higher value of cohesion in the visual mode. This is the visual mode over here because we are forming closely packed clusters over here. So to take home message over here is that the sensing modalities have a key role to play on how the group level behaviors would be. At least that's what we can learn from this simple ideal simulation setup. Okay, now finally, I would like to go over what we started with the experimental data of bats, how we can test these information theoretic tools to see if we can have the same success with the simulation data where we know the ground truth. That's going to be the final topic over here. And in the simulation setup, I generated a scenario where I have, this is the field of vision, again, rear to front. So this is the rear guy can see only the front guy, and the directionality of interaction would be from front to rear because the front individual cannot see the rear individual, whereas the rear individual can now see the front individual. And again, I run the simulation, generate some trajectory data, and from that trajectory data, I compute a curvature-based time series. Also, I conducted an audition-based sensing where now they are using this acoustic beam where the front guy over here is residing in the acoustic coverage of the rear guy and can listen from the rear guy. So the directionality of interaction in this audition-based sensing is from rear to the front individual. So that's what we did. And again, we generated some trajectory data. We computed curvature, just like the way we did it for the experimental bat study. And then this is the results for the transfer entropy for both the auditory and the visual mode. So we ran it several times, like we conducted a Monte Carlo simulation. We generated a multiple trajectory and implemented this transfer entropy. And just to give you the main message over here, transfer entropy was successful in identifying the correct directionality. For example, in this auditory mode, it identified the rear to front coupling, which is over here. In the visual mode, it identified the coupling direction is from the front toward the rear. So this shows that this allows us to test the data analytic tools in a simulation setup where we know the ground truth, we can generate and we can validate the tools. So what we learned today. So I'm going to having some time to do the test to show you the example how to do the use the JIDT toolkit. So just to summarize, what we learned today is the importance of the data analytic tools that can allow us to extract the features of the real world interactions. And we also saw a lot of different diverse applications of these data analytic tools. We also briefly talked about a model, a very simple model that we can use to study collective behaviors, where we can also modify that model to incorporate different types of interactions. For example, this model has been modified for more realistic scenarios where particles are moving in 3D and they have like more noise and their particle avoidance, like collision avoidance, those kind of stuff can be implemented. We also talked about how different metrics we can use to quantify the group level behaviors. For example, the order parameters, the polarization, which measures the overall group alignment, and cohesion, which measures how closely packed the group is. And we saw that how we can take this simple model and use its power to implement different sensing modalities in a very idealized situation, for example, a vision-based and audition-based. And the learning outcome from here was to show that these two sensing modalities play a key role in how the group level behavior would be for some choice of sensing parameters. Then we talked about the IT tools, which we can also kind of validating these information theoretic tools that can distinguish the sensing modalities. Okay, so that's basically covers the more or less my presentation. The remaining time, I'll try to show you an example of how to use the JIDT toolkit. So before that, let me ask you if you have any questions. Let me open the chat window. You can post the questions over here. Thank you, Amit. Thank you for the nice presentation. Any questions that you may have? Okay, so let's, if there are no questions, let's go to the demo. For the demo, we're gonna consider this I just wanted to generate some data where we know the ground truth, and then we can show how we can implement this JIDT toolkit to identify the coupling strengths. So to do that, I chose like the logistic map with two different couplings. For example, beta xy is how much y would influence on x, which I chose to be 0.15. And here beta yx means how much x influences y, which I chose to be 0.5. So it's an example of unidirectional coupling where, or it should be like dominant direction of coupling is from x to y. And we're gonna see whether JIDT toolkit can capture this. At least it should be able to point out that there is a stronger coupling from x on y compared to the other direction. To do that, what you have to do is, thanks, Brooke, for sharing the link. So you can go to this link over here and click on the Google Drive folder. And if you click on my name, it has the resources that you can download. So this is the Infodynamics toolkit. So before I show you how to download, let me also show you the GitHub link that I shared with you in my presentation. So this is where you can find the JIDT toolkit. It explains how to install, download, and installation. It's very easy. You don't have to do basically anything. You just extract the folder and you have a nice GUI, which is ready to use. You can load the data and start. And there also, it provides some documentation, and there are a link of courses somewhere over here, where it takes you to this series of lectures, which introduces to this concept of transfer entropy and conditional transfer entropy. So for the download, if you click the link, you will come across this, the zipped folder, which you can, again, click to download. So I have already done that over here. So you will see this is a folder. I'll take some time. So if any of you are trying to do it with me, so I can wait. So this would be the Infodynamics -dist -1.5. This is the version. And maybe this is the most updated version. This is what you have to use. And once you download that, you have to extract it, and you will see a folder like this. Also within the folder, you will find some interesting things. There are some tutorials. It also has the README tutorial and exercise, which are very helpful if you are new to this topic. And there are also, this is the JIDT explains all the parameters. I only gave you the very top level view of how this transfer entropy works, but all the details of the parameters are provided in this PDF. So once you have that, if you have already extracted this folder, you will see within this folder, there is a Java application. It's called Infodynamics. If you click on this, it's gonna pop up and ask you for some options. You have to provide which calculator you wanna use. So these are different information theoretic calculators, depending upon what you need. For example, in this case, we have 2 time series data. I just showed you from the logistic map that we have generated. And we'll also find in the Google Drive folder, unidirection.txt. This is the data I've already generated and saved it for you. And if you download, you will see this is the text file. So it has 2 columns. One is where the first column corresponds to X, and the second column corresponds to Y. So now how to load the data? You choose transfer entropy because we have 2 time series variables. It should expect 2 time series data, and it's gonna give you some options. So this window would pop up, and here you can choose different type of calculators for the PDF estimators. So when you have the sample data, you have to estimate those probabilities. So there are different types of estimators. I would recommend using Kraskov, which has the bias corrections incorporated in it. And you can also use the discrete or kernel-based estimator. Here, next is where you can load the data. If you click on it, it's going to ask you to give you the right directory. So I have it in my downloads folder. So let me see. I've never done this in this way, but yes. So here is where the downloads folder is, and I have the data unidirectional.txt. I'm going to open it. It automatically going to show you that how many columns and how many rows are there in that file. So now I have 3000 samples and 2 columns, one for corresponding to X, another corresponding to Y. The third option is it asks for like whether you want to compute the transfer entropy from X to Y or from Y to X or from both the directions. So let me start with, I'm going to click it all pairs later, but let me start with the source and destination over here. Source and destination. So this is 0 because here the indexing starts with 0, unlike in MATLAB, which starts with 1. So it's 0 over here. That means we are trying to find how much X influences on Y. And the destination column is 1. So these are the statistical significance tests. If you click it, it's going to also conduct a statistical significance test. I did not go into too much details of this because of lack of time, but basically you generate some surrogate distribution where the sequences are disturbed, and you conduct this conditional transfer entropy or transfer entropy, whichever metric you want to use, on those surrogate data, and then compare with your actual measured data and conduct a right-hand tilt test to see whether it's within the 95% interval or not. So that's what exactly it does, but you may skip it for the time being. And here is all the parameters that you can control. In the presentation, I showed you as a first order Markov process, but you can have it as a higher order. So you can change the parameters over here, K history. But just let me show you what it happens if I click on this button over here, generate code and compute. It provides me the transfer entropy value. It shows that we have used the Kraskov estimator, and from X to Y, it has computed a value of 1.2627 natural units, so log base E. Here also on the right-hand side, you would see there are different scripts that has automatically been generated. So I'm a more of like MATLAB fan, so you can copy this part and go to your MATLAB, open a text, and just open a new script and copy it over here. So it should be perfectly in working condition. It's going to give you the same value. So that's a nice feature. You can generate the script over here. Once we hit the all pairs, that means it's going to compute the transfer entropy from both the direction. It's not going to show any results, but it's going to generate the code for you, and you can copy this and again go back over here, paste it in the script, and let's run it. You see it returns 2 values. So as I said, transfer entropy generally are 2 asymmetric quantities. Here from X to Y, it identifies the coupling strength is or information flow is 1.26 compared to Y to X, which is 0.43. So the tool correctly identifies in this case that the correct or dominant direction is from X to Y, and there is also coupling from Y to X, but it's less compared to X to Y. So that's basically it for me. I think there are a lot to explore. I couldn't cover everything, but here are the resources again that you can use. If I go back to the GIDT slide, this is the tool. Like this is the GitHub page that would be pretty useful for you. And listening to the courses, this YouTube videos would be very helpful to get accustomed with this concept. With that, I would like to stop. Any questions? Glenna, if we have time, can go over what is in each column of your sample data. Right. That's a good point. So this is the logistic map. Let me go over here that we use to generate some data. So it's a discrete map, and we started with certain initial condition. I don't think I have the script with me here in this folder that generated the data, but it's basically this is the X values with certain initial conditions, and then these are the Y values for, I think I ran it for 5,000 time steps. Any more questions? Or questions could be asked for me or any of us. So we're all, it's more of open.
I guess I could ask a question. You started your presentation at the point where it's kind of after you've collected the motion data already, if you were trying to apply your information theoretical framework to data that's already been captured. Do you have any specific recommendations for how you can optimize the experimental setup and the data collection to make this analysis work as well as possible?
So I was not the part of the data analysis group. It was mostly an undergraduate and group of like a master student who worked on this. So if I remember some of the details is like we used 6 cameras, and those were like with GoPro cameras, and we used some infrared lenses that we replaced in the GoPro cameras. And again, like most of the stuff that Glenna mentioned of is how to synchronize the cameras, that was immensely challenging. But the most problematic was that when we had this frames by frame tracking each individual, they're not like a lot of pixel resolutions. One software we used is called Blender. So Blender, like last time when we saw it, it was like 3-4 years back. It can automatically track for between frames for a given pixel. That reduces some of the like manual effort that would be required to tracking those. So it's been a long time, but mostly I was the data guy. So this is all I remember.
When you're tracking collective behavior, do you have a lot of problems if individuals are kind of going out of the field of view and back in, so that you're not always tracking the same individuals, or does that not really matter too much?
Right, that's a good question. So we had like 30 pairs of bats that we tracked. So there are different individuals, but we conducted our analysis on like 30 pairs, and we had different control conditions. So one is where we do not have any obstacles, and the other conditions where we provided some like obstacles in terms of thin films, so that we can see in this testing environment whether the behaviors are different, which gives us a basis for the comparison. Going back to your question, it was not the same individual, but it was like different pairs of individuals, and that was a challenge because information theoretic tools rely on this large amount of data, and we had to like concatenate all these pairs to identify the distributions. So there are different approaches nowadays. We can also conduct like for each experimental setup can be tested in a different way. Like the JIDT has some features that has been now incorporated where you can provide data from each experimental session. But at that time, what I did was like concatenating the 30 pairs so that we can answer this research question.
I see, thank you. Do we have any other questions from the audience? You can enter things into the Q&A or directly into the chat, or raise your hand. There's a question in the Q&A window. See if you could.
Yes.
Are there commonly used databases? Sorry, go ahead.
Right. So are there commonly used databases where people upload animal data? That's pretty challenging. Like, where do you find the real world data? At least for us, for the bad data, we didn't have from like a lot of data available to answer this research question. So we had to go to field and collect the data. But this is something I know there are some initiatives have been like devoted to collect data from different research groups, for example, starlings, bats, fish schools, that their efforts have been put to make a common databases. So I think it's not published yet, but from our PhD group, there has been some effort to collect all the data.
Yeah, there's no like single repository, unfortunately, but there are, I think, a lot of papers that publish their raw data these days to online, like Dryad kind of stuff. So yeah,
we did, like, we shared through a paper like all the data. That's probably would be useful.
We have a question from Jill McRae in the chat window.
So the question is about whether the resolution of the head body information from the cheetah example could be used in the flock dynamics. Unfortunately, Amir had to step away. So in terms of speaking about the resolution, but maybe you could talk about the dynamics portion of it.
I'm interested if the resolution of head body information in the cheetah example can be used in the flock dynamics. You mean like the finding the coupling relationships? I don't think
I guess
I'm
it
reading
would
the
have to be
question.
that because there's only one animal in the field of view, so it would have to be about the interaction between the head and body.
Right. So it's basically like when you have some form of time series data, if you're trying to come up with like identifying the relationships, like any kind of coupling direction that we're trying to understand, probably that, but I'm again not sure. Like it's a single individual.
Jill says yes, to go beyond moving dots to what the body is doing.
Yeah, I think I think when often looking at one individual, I haven't seen as much like use of information transfer for kind of getting from individual dots to body movement. I've seen more of like applying skeletons or looking at kind of joint angles based from the moving dots, that kind of stuff, to kind of get more at the full body motion, but haven't seen so much about using information transfer for that, more for within individuals.
That is an interesting thought, though, because it might get, rather than at the mechanics of the body, at potentially for what might be some of the sensory channels that are being used, or the directionality of the sensing information, at least between the head and body.
That reminds me a nice work from the Joseph Ligier itself, like where he used a robotic, like a snake, and he measured, like used information theory to find the, like again, the coupling between different parts. So there has been some, I'm not sure, I did not go into too much, have not explored this topic for a single individual yet, but there might be some interesting study.
And there is some work done on that for sure in human work I've seen a little bit, but it hasn't so much been information theory. There's been more like fast Fourier transforms to see what the offset is and how much, like when it's influencing and when it's not, kind of like kinetic chains of like which joint is influencing the next kind of joint stuff. So yeah, I definitely think there's a lot of potential for that.
It strikes me one organism that this could be particularly interesting to apply it to is animals with a more decentralized nervous system control. So like sea stars, where you have all these tube feet, and you could try to understand the interactions between them. Yeah,
that's a cool idea. Like there are complementary aspects that we have presented today, so that can be a cool integration. Definitely, I see there's a comment on that.
All right, we are going, past 3 hours, I think, here. So maybe it's time to wrap up, unless anybody has any additional burning questions or if you have closing remarks. I want to thank the speakers. I really appreciate the tools that you've provided. You've done such a nice, thoughtful job of providing some accessible materials that people can use to go and learn about this in more detail after the workshop. So I really, really appreciate that. Thank you so much.
Thank you for the opportunity.
Yeah, thanks so much for organizing, and feel free to reach out if anyone has questions. So
yeah, sure.
All right, the recording will be posted online on the SIMS website, along with all of the materials that everybody's gone through during their tutorials today. Thanks so much.
Perfect. Thank you.